Welcome back, everyone, for day 3 of our conference. Before a reminder, just to check the expectations that are in your conference program. We went through them in day 1, but we've asked everybody to just kind of go back through them on their own in the conference program and just remind ourselves of kind of why we're here and how we're going to be working together today. Today's theme is collective deep thinking in ME research design. We hope that this theme will help us to use everything that we've learned so far. So everything we learned on day 1, day 2, and then we're going to learn some new stuff today as well. And we're going to try to put it all together by the end. We hope that it's going to help us inspire us to reimagine how we can design research that is more inclusive, accessible, safe, collaborative, informed by decades of knowledge, our historical context, and strong, knowledgeable patient partnership, to name just a few things that we're going to try to do. As you can see, we have an ambitious agenda for today. We have some pretty amazing speakers. A few of them you'll recognize. They're back for more. And we hope it'll only be the start of more discussions like this. So please join us in welcoming our wonderful speakers for today. And we're going to get started with Todd, who's going to do a session on critical thinking in research design. And he's going to walk us through some pretty important things that we've been talking about a lot in certain circles, Todd. You're going to bring everybody into that circle, and we're going to kind of hear some thoughts on what we could be doing in that research space. Thank you so much for being back. Oh, you're on mute.
How long have we been using Zoom? I still have not figured out the timing of the mute button. As I started to say, I'm privileged to be back. And as I told the group before we came on, there is no higher compliment than being asked to be back and not getting a text after the first talk saying, well, maybe we decided to go a different way with this. So hopefully you are seeing my slide and not my speaker notes.
Yes, we
Sabrina,
are
is
seeing
that
your
correct?
slide. Yep.
Awesome. So the planning committee, thank you so much again for having me back, and thank you to the attendees for tolerating me for another half hour here. I was asked to do some level setting on day 3, pulling together threads from day 1 and day 2, and introducing the day in which we'll be talking about, as Sabrina mentions, really deep thinking about how to co-create, how to co-design, co-analyze, co-disseminate research studies in this ME space. And so we'll go ahead and do that. Again, the standard disclosures apply. These are my affiliations and my employment. I have no financial or non-financial conflicts related to this talk, and I'm speaking for myself, not for my employers or affiliates, but they might share it on social media if they like it. So here we are with our agenda for today's talk. We're going to talk a little bit about the current status of ME research, where we are, kind of a clear-eyed assessment of the current landscape. And then I'd like to organize this talk around 3 propositions of organizing research in ME around understanding post-exertional malaise, a baseline consensus for underlying assumptions that we use in ME research. I'd like to then get into some of the more kind of nerdy technical nuts and bolts of sampling and validation as the 3rd proposition, and then just some concluding remarks about designing better studies. So where are we? It's a very clear-eyed assessment of the current status. We have a rapidly increasing global frequency of ME. We have a poor and now often declining source of research funding, and I'll also add clinical funding. And yet we have no approved tests, no approved biomarkers, and no approved treatments. The funding landscape of ME has sort of always been challenging for people wanting to enter the ME space. This is work done by Miran and colleagues that identified that not only is ME significantly underfunded with regard to other National Institutes of Health funded diseases in the United States, but it's also significantly underfunded even among underfunded diseases. So what this means is no pipeline of new ideas, no pipeline of new talent coming into the field. And so this is a very under-resourced area of research traditionally. Now, the emergence of COVID and long COVID has changed that temporarily. You may have heard of the Recover project and the Recover TLC continuation of that project that is focusing on clinical trials, where here in the U.S. we've spent over $1 billion on trying to understand COVID and long COVID, and largely most of that went to observational studies of epidemiology. And so, it's since become clear that funding will become a lot harder to come by. The current political situation is not helping us here in the U.S. Related to funding for ME and ME-like conditions. And so things are very much at a standstill. Long COVID is an important source of new cases of ME. And before people get upset with me for saying that, let me just sort of reiterate that I don't think that all long COVID is ME, but I do think that there is ME within long COVID. And we know this because post-exertional malaise is present in about a quarter of community dwelling adults with long COVID, and that the incidence rates in post-COVID condition of ME are significant, are about 3 times what we see in people uninfected by COVID. So maybe the way we think about that is the way we've always developed ME. So as it turns out, SARS-CoV-2 is an important driver of new cases of ME/CFS. And so here we have an increase in frequency of ME, and we've spent all this money, and we still have very few answers to show for it. So against that hopeful start, I would really like to get into how it is that we optimize our use of resources and make sure that we are doing the best we can to develop the best information possible for people living with ME. And as I mentioned, I'm structuring this around 3 propositions. The first one is the lengthiest, and it really tags on to my talk from day 1, as well as pulls from threads throughout both days of the conference. And that's to say that because post-exertional malaise is central to the lived experience and diagnosis of ME, it always should be central to how we design and organize research studies about ME. In its multiple forms, the multiple forms of research that we do, diagnostic studies, treatment studies, all should be focused around post-exertional malaise. So what does this mean? Well, we'll start with what's post-exertional malaise, and as I mentioned, we don't define post-exertional malaise necessarily by post-exertional malaise. We use post-exertional neuroimmune exhaustion, and that's characterized by the features you see on the screen, which are marked rapid physical and cognitive fatigability in response to exertion, post-exertional symptom exacerbation. So in other words, symptoms become signs and symptoms are worse after exertion. Potentially progressive exhaustion that is delayed after an exertion with a prolonged recovery period of a few days to a couple of weeks to sometimes never. And then this low threshold of fatigability that frequently becomes lower over time and can cause progressive functional deterioration. So Todd Hunter Brown and colleagues did a really interesting study that came out in the spring of this year, looking into about 680 different research studies in ME research between 2018 and 2023. So these are recent studies. And they looked into various different characteristics in order to map the evidence. And it's a massive piece of work, and we won't get into the whole thing, but we'll pick and pull from parts of it that are relevant to this talk. And certainly, if you want to get together and discuss the rest of it later, I'd be down. It's a very good study. One important piece is the case definitions that are used in ME research. And again, remember, this is recent stuff. And notice that the Fukuda criteria on the far left here are still, by far, the most common as far as the criteria are concerned. The next most common, and I think you can see my pointer, is this category of not stated. So didn't even have a category, maybe just nominally referred to something. And then you have this other massive category of other. And so before we get into these top bolded studies, you have a massive number of recent studies that either have used criteria that don't require post-exertional malaise, or we don't even know what criteria were used. And so, the Canadian consensus criteria, the international consensus criteria, these National Academy of Medicine criteria, the UK NICE criteria, all require post-exertional malaise. But it just gives you an idea that we can't even have consensus on including PEM as a research criterion at this point. And I think that would be a very basic thing that we need to do. And just to say that even if we agree on a case definition criteria, this is data from Lenny Jason's group, that each of the case definition criteria identify a slightly different cohort of people living with ME. So even within these criteria that are much more specific that require post-exertional malaise, we need to come to consensus on which of these we'll be using, or be aware of the selection bias associated with the case definitions. So why should we do this? I mean, first, it starts to correct the course of an entire history of injustice, bias, and gaslighting. I cannot wait for Dr. Fung's discussion of this later in the program. It also provides us with more fidelity to the lived experience and disability of ME, which in turn enhances the validity of the research findings and generalizability. And then finally, it allows us to come up with PEM-compatible study procedures, and this may help us to improve inclusion and accessibility of the research, of participating in research to more individuals. The cautionary tale, obviously, is the UK PACE trial. We're well beyond a conversation about this in terms of our time today, but suffice to say there were a lot of problems with this study. It is still stuck in the literature as level 1 evidence. People still cite it as evidence of effectiveness in so-called chronic fatigue syndrome, and extrapolating that to ME. But we also know that it was based on the Oxford criteria, and the Oxford criteria are not specific to this ME cohort that we frequently discuss. So it does raise the high likelihood that people who are deconditioned, depressed, and so forth, who can respond to exercise, were mixed in to the study cohort. And so this is obviously a major cautionary tale. But also remember, the evidence that's out there suggesting that the effectiveness of exercise reduces as we start to include more rigorous case definition criteria involving post-exertional malaise. And so our very results will depend on the people that we enroll in these studies. And that also focusing on post-exertional malaise has clinical importance, that we improve satisfaction, we reduce the likelihood of deterioration if we focus on post-exertional malaise. So our research in ME should center post-exertional malaise. But what does that mean? So we start with this whole idea of PEM being all of these signs and symptoms and comorbidities that we've talked about, and many of these are obviously horribly disabling. It's a very individualized experience. Each of these are affect different people in different ways, and may be present after different exertions. Some of these comorbidities may attract researchers into the field based on something that they recognize from their own research background as being potentially at play in ME. And I would invite us to really remodel our thinking around post-exertional malaise. So what does this look like? Well, we start thinking about the body's inability to respond to an exertion, taking it out of homeostatic balance, and really sort of thinking about the fluctuating nature of signs and symptoms, the disability that they cause, the individualized science clustering of signs and symptoms, all based on this post-exertional characteristic as an organizing feature, instead of thinking about it as sort of just an extra thing. Some people get really concerned that maybe now we're conflating different conditions. And so I would just remind us all of Hickham's dictum, which is sort of the opposite of Occam's razor, which is to say that a patient can have as many diseases as they damn well please. And obviously that's overstated. Nobody chooses to have a disease, but this is just to remind us that we need to take an expansive view of these signs, symptoms, and comorbidities, and avoid premature closure in our thinking about causation. And so again, Hickham's dictum should really influence our thinking there. And so, as I mentioned, long COVID is an important, SARS-CoV-2 is now an important new source of cases of ME. And in fact, when we look at people with long COVID who meet Canadian consensus criteria that select for post-exertional malaise, they have greater functional impairment, they have more frequent reports of impaired recovery responses, and they have more frequent reports of worsening with physical and mental effort, suggesting that identifying ME, even in nominally non-ME studies, becomes really important in order to ensure sample uniformity and to enhance the validity and reliability of our results. So, as we start thinking about exertion, it's in the name, post-exertional malaise. The most inclusive studies will use decentralized data collection and interventions. That allows us to ensure that we include a range of disease severities and access to resources and so forth. But the most reliable studies historically have used post-exertional paradigms. They either control or induce exertion in order to measure the post-exertional response. These measures tend to induce PEM. And so there obviously are the advantages that you have a measurement to actually measure what's happening in the post-exertional state, the disadvantages that you're inducing PEM, obviously, and that's a huge one. But interpreting single measures is always going to be less reliable than interpreting repeat measures. And we have to think about the confounding effects of pre-resting, pre-fatiguing, even in decentralized designs where we need to give very specific instructions to our patients about when and how to take those measures in order to ensure that we're not introducing important confounding effects of exercise or other self-management tools like pacing. People may be more likely to pace in the treatment arm than the folks that are just being watched. And so here you have sort of this confounding effect. The other thing to point out here is that studies that use exertional paradigms should use the minimum necessary exertion, and that submaximal isn't often gentler. It's not often more PEM compatible because people are exercising at a low level, exerting at a low level for a longer period of time. So as it turns out, the exercise dose or the exertional dose tends to be as great or greater than maybe a more acute exertional paradigm. So just remembering that submaximal doesn't always equal PEM compatible. And that we do have to quantify the dose to understand the effect of the poison. So, all of the other sort of submaximal types of exertion still need to be validated against the better characterized, better historically characterized sort of exertional paradigms, because you get a chance to measure the specifics of the exertion. So again, as we start thinking about biomarkers, as we start thinking about alternate forms of exertion, we can't forget about what we've known before, but we should build. We should use that as a foundation in order to build. Evaluating for post-exertional malaise is science and art. The current recommendations from the U.S. National Institutes of Health suggest initial screening and then a confirmation step. The core PEM assessment questionnaire you might recognize as the DSQ PEM, and then some additional screening questions that have been created for that purpose. And so the scoring of step one to include a person with potential post-exertional malaise is that they need to have a severity score of 2 on at least 2 of the first 5 items, which just equates to at least moderate impairment half the time. And then items 7 or 8 must have an answer of yes, along with a prolonged response time. So that's step 2. And so this then gets people into consideration for post-exertional malaise. And so why does this matter? Well, if we just looked at step 1, the step 1 screening questions might not be sufficiently discriminative of people living with PEM versus, say, people living with post-polio syndrome and multiple sclerosis. And so you have to also screen using the supplemental items involving exacerbation with low-level activities and impaired recovery. So if we're just using those first 5 items, we're missing potentially something very, very important in that. And just to support this, I did some calculations for you, so I apologize. This is a really busy slide. But I just wanted to point out that these supplemental questions tend to be more sensitive than specific, which is great because this is a screening questionnaire, but they're most effective when they're negative. And so when we use this questionnaire where we're asking the participant to endorse the positive items, actually the clinometric properties don't shake out quite as well. And so using the DSQ-PEM as an outcome measure is a little more challenging, in addition to how it's scaled and so forth. But just remember that the tool is very strong for what we use it for, which is screening. But to use it as the gold standard to identify PEM in research studies is not altogether consistent with the National Institutes of Health recommendations, and the use of the DSQ-PEM to measure disability associated with post-exertional malaise is not yet validated. So, and maybe not supported based on the clinometric structure of the questionnaire. And there are other problems with other fatigue-specific outcomes measures. So surveys to screen for PEM aren't necessarily appropriate to measure the disability associated with it. Common fatigue-specific measures are not sensitive to changes in induced PEM. And we also know that measuring single symptoms doesn't necessarily maintain adequate fidelity to the entire lived experience of post-exertional malaise. And so we should use measures validated for purpose. The FUNCAP, the PAC are 2 examples of 2 post-exertional malaise specific kind of condition-specific questionnaires. There may be others as well, but again, to be very intentional about how we're powering studies, calculating sample sizes, and understanding the functional effects of the lived experience, but also of interventions. So we'll move on to proposition number 2, is that we should really think about ME as a principally biological condition with potential secondary effects that are psychological and sociological, and not the other way around. So we've seen this word biopsychosocial that tends to be operationalized as all caps psychosocial. And I would invite us to consider all caps psychosocial. As Maya Dusenberry so eloquently took us through the history on day 1, there's a common root in sort of ME and FND in neurasthenia, and sort of the road path departed in the wood as the infection associated cases became much more apparent in the 30s. And then the pathobiological disease construction really sort of took root from that point forward. Whereas the more Freudian construction on the bottom went through to various different iterations, largely unchanged to the present day. And so the diagnostic criteria for FND remain a diagnosis of exclusion. And by what I mean by diagnosis of exclusion is that third bullet, which is that the symptom or deficit is not better explained by another medical or mental disorder. And of course, ME is replete with pathobiological evidence that we can measure reliably that can be used to explain post-exertional malaise. So on its face, post-exertional malaise, and an ME is not a functional disorder. It's not to say that people with ME can't have functional disorders, but that as a matter of course, we should not consider ME as a functional disorder. But there are plenty of conditions that are commonly misdiagnosed as FND, autonomic conditions, chronic pain conditions, autoimmune conditions, genetic conditions, different mitochondrial and metabolic conditions, and then infection-associated chronic diseases. And so just to kind of bear that in mind, that the FND label really doesn't work here. And that we can differentiate ME and long COVID and FND based on a clinical exam. And many of these clinical exam items serve as the basis for identifying cases of ME in research. And so this is also worth pointing out. My third proposition is that we should plan ahead to ensure treatment is available through research and to validate subgroups we anticipate that we might find in research studies on diagnosis and treatment. And so there's a few items in here. You know, the ethics of a control group are sort of questionable here because we're ultimately denying care for people who are historically denied care. And that sets up an obvious problem, even though the logic of science suggests that we should give a person treatment and the other person a modified treatment or no treatment at all, and we should compare them in order to establish cause and effect relationships. And I just want to shout out some current work doing with my colleagues with Renegade Research, two of whom we'll hear from later in the program, on crossover design. So in a randomized crossover design, you take a group of people, you split them in half, you randomize one to receive treatment, randomize the other to a control, and then you cross everyone over into treatment so that everyone is treated. And then taking outcome measures along the way. And the advantage here is twofold. First, everyone gets to try the treatment. No one knows when they'll get the treatment or how long they'll have the treatment, but everyone will get the treatment. And the second advantage is that you get to assess questions about the duration of treatment, maybe the different levels of dosages of treatment as you go along. So you can answer multiple research questions with this single, really elegant design. You know, and of course, there's some trade-offs here. You're treating people now potentially for twice as long, so you have to plan for that. It takes a little longer for results to come out. You have to plan for more of the intervention and so forth. But there are some real advantages here in order to resolve some of the issues of injustice related to controls. You know, we really hope to be able to differentiate between two groups in a research project, much like we have, you know, sort of on the target. So in the red, maybe we have all of these folks who received a treatment and a beneficial treatment effect, and those in green or maybe didn't have the treatment and don't have that beneficial treatment effect. But in ME research, we frequently see this, where we have maybe a couple of people who got a beneficial effect and a couple of people who are clustered around another area. So it brings up this idea that we need to subset or we need to subgroup. And so maybe we just move the target over and analyze what does that group look like? And maybe we then well, my animation's not working, so I apologize. Maybe we move over to the other group and we try and move the target over to the other group. And I just want to point out that if we do this approach, we should plan ahead. That's going to require a couple of steps that are important. We need to characterize the predictive characteristics when we talk about a cohort or a subgroup, and then we need to validate that subgroup using a separate sample. So saying there's maybe a subgroup is a good start, but it's not good enough. It does not get us closer to understanding in who will respond to a treatment or who may be harmed by a treatment. So planning ahead to do a thorough, careful subgroup analysis becomes really important. And then finally, we have a whole host of equity issues related to participant recruitment. So the map that you're seeing is from the Todd Hunter study. And you can see that most of the studies are conducted in the US, Canada, the UK, the EU, and Australia. Very little from entire continents that they're underrepresented. Their health systems, their patients are totally underrepresented. Only 19% of studies published after 2018 presented on severity. I apologize. I put age, I meant severity. Mild in about half, moderate in about 60%, severe about 70%, but very severe are very underrepresented in the literature. So again, these decentralized trial platforms get us closer to the inclusion we need. About a fifth of studies reported on race, but those studies were about 95 to 99% white. And white people are not the only people who get this disease. We need to do a better job of including people from across races and ethnicities. Children were only represented in 10% of the studies, and there were no studies solely of older adults. As people get older with ME, we have no idea what the effective age on ME is as it relates to that. We need to do a better job of including participants from across the lifespan. So really our opportunities are many of that we've heard. We need trainee development and training opportunities for established scientists that are crossing over into ME research about post-exertional malaise. We need to have lived experience experts who can guide the creation, the analysis, the interpretation, and the dissemination, including patient partners and engaged caregivers. We need to be committed to inclusive, accessible, and equitable research designs and platforms. And we need to carefully characterize and cluster cohorts and subgroups based on signs, symptoms, and comorbidities, but we can't stop there. We have to replicate those clusters in order to ensure that we're not, what we're seeing is not an artifact of mathematics and sampling bias. So in summary, post-exertional malaise is central to lived experience, and so it should be central to our research. We should require PEM in case definitions. We should go through the correct painstaking steps of identifying PEM. We need to account for exertion. We need to consider ME as a principally biological phenomenon while we promote inclusion, accessibility, and rapid validation of our findings. Thanks.
Thank you, Todd. That was a great start to our day, a great warm up for us to get started and thinking about all of these. I know there are some questions for you in the chat, Todd, so if you wanted to go in and try answering some of them, that would be great. And we're going to move on to Jacqueline Becker, who I'm just going to say, Jacqueline, that I didn't understand, and I've said this to you, I didn't understand what role you guys could play as a profession in our research before I started working with you on the NIH committee, you and the team. And you guys have completely turned my mind around and helped me to understand how we could be doing this better and getting better results for patients, and thereby then putting us in a better position to be able to get better treatments. So I now understand why we should invest in this. So I'm excited for you to explain to us a little bit about what we could be doing, and maybe we could have you back for another webinar another day where we delve a little deeper, because I think this is a big topic we need to learn about. But welcome, and you can start whenever you're ready.
Great. Let me just share my screen here. Let me see how I can do this. Okay, can everyone see the main slide?
Yep, that's perfect.
Okay, great. Okay, so hi everyone, and thank you, Sabrina, for that introduction. I'm so happy to be here with you all today. I'm a clinical neuropsychologist and a health services researcher at the Icahn School of Medicine at Mount Sinai. And my research has largely focused on chronic diseases and cognition in chronic diseases, but specifically in infection-associated chronic conditions, and more so on long COVID. But we've been working with a lot of ME patients and doing a lot of work in this space as well through the NIH. And so that is where I met Sabrina, and I'm so happy to be here. My talk here today is really geared towards people living with ME, and specifically for those of you living with some of the cognitive challenges that we're going to discuss today. And my goal is really just to give you some insight into how researchers and clinicians think about cognition, and so what it is, how we measure it, and importantly, why the tools we have sometimes don't always capture what you're experiencing. And ultimately, my hope is that helping gain a sort of better understanding of these concepts can also help you advocate for yourself and help us build better measures and better studies for the future. So I'm just going to start my talk. Oh, I can't. Oh, there we go. By talking a little bit about what we mean when we say cognition, right? So what is cognition really? When we talk about cognition, we're talking about the thinking skills that allow you to sort of navigate the world and carry out, everything from basic to complex tasks. Many of you know these terms intimately because you've noticed changes in one or more of these areas, but it's also important to understand cognitive ability as being sort of a parameter or a marker of integrated function of the brain. And this is because these cognitive domains and subdomains that you see in these blue boxes here, they don't work in isolation, meaning that for one domain to work properly, it often recruits other domains as well. So, for example, in order to have memory, you must first have attention, right? So you have to pay attention to the information that you're trying to learn. And attention is not just your ability to focus on the information presented, but also on your ability to filter out distractions. And so that's why there are so many subdomains of attention as well. And similarly, your processing speed must be intact, right? You have to quickly take in the information before it sort of leaves your mind. You need language, which involves not just understanding and producing verbal information, but also your ability to assign meaning to words, visual-spatial abilities if the information is presented visually, right? And so you need all of these different domains, and they all sort of work together. And one of the most important ones that we like to think about too is executive functioning. I like to think of executive functioning as akin to like the CEO of your brain. It sort of multitasks and organizes, it plans, it problem solves, it helps you initiate behavior, it motivates you. And so that's sort of how we think about executive functioning. And so if, for example, the information that you're trying to remember is disorganized, or if the information, if you're having difficulty with things like abstract reasoning and understanding the information in very abstract ways, that can make it difficult for you to remember. And so oftentimes people say they feel forgetful or may report memory problems when really the impairment may lie in another domain, like in attention, executive functioning, processing speed, etc., rather than with the true storing of information. And this is why, when we measure cognition, we really have to measure all of these domains. And so in conditions like ME and long COVID, people tend to fatigue really easily. And so a lot of people will come in and say, well, why do we have to look at every single domain and do all of these different tests? It's just my memory that's bad. Why can't we just test memory, right? And so this is a little bit of why. And then this brings me to how we actually measure cognition. The way that we do it is we use standardized tests. And this means that everyone has to take the same test under the same conditions, but that doesn't really tell the full story, right? So, if you see in this cartoon, it says for a fair selection, everybody has to take the same exam, please climb that tree. And so for those of you who can't see the picture, it's essentially a bird, a monkey, a penguin, an elephant, a goldfish, a seal, and I think that's a goat at the end. And so essentially, that's not a fair test, right? To have all of these different animals, some of which are obviously much better at climbing trees than others. And so what we do then is we take each individual person's scores, so what we call their raw scores, and then we compare them to people of similar age, sex, and education to make it a more even playing field, so to speak. And that's better, right? But that doesn't tell the full story either. And this is because when we think of cognitive test scores, they typically fall along a bell curve where most people cluster around the middle. So sort of that line right there as considered sort of average. And we usually consider impairment as falling 1.5, so 1.5 standard deviations below that normative mean, that average line there. But the reason that normed scores don't tell the full story is because not everyone starts in that average range, right? So if you are functioning in the high average or superior range, so maybe you're a little bit above average, right? That same standard deviation won't apply. And so, we sometimes will have people who were previously surgeons before they got ill, or lawyers, or extremely high functioning before they developed their illness. And so when they're scoring in the average range, usually we figure that represents a significant decline from their personal baseline. And that's why when we look at your scores, we also try to estimate your premorbid ability. So premorbid ability is your likely level of functioning before your illness. And you do that by looking at education, prior occupation, but we also have specific tests that correlate with your sort of baseline IQ. So tests that represent crystallized abilities that are not meant to decline over the course of your life. And so really, that would allow us to give you sort of a personalized look at what might be a decline for you. So, you may still be in the average range, quote unquote, but again, it might be a decline for you. So that's one of the reasons why sometimes people will go for a neuropsych evaluation with complaining of cognitive problems, and the results are that they're normal or average. And then another, particularly challenging thing about cognitive symptoms is that they're often invisible to others, right? So again, someone might look at you and think you seem fine, but inside that your brain is sort of working 3 times as hard to do something that used to be automatic and that is automatic for most people. And unlike fatigue or pain, cognitive symptoms can also sometimes be really difficult to describe and difficult for other people to understand. And so this is why measurement really matters. We need tools that can capture what you're experiencing, validate that it's real, and track whether interventions are helping. So, there are a lot of people in the community that feel like, we should just throw away, objective cognitive measures altogether because they just classify people as average. But I think the issue with that is that throwing these sort of tests away does a disservice and doesn't allow us to sort of work and try to figure out the most ideal tests, which is part of what we've been doing with the NIH, is trying to figure out which tests are most ideal, are most reliable, valid, sensitive, and specific, that can actually detect real world problems. And so a good test in general needs to be both reliable and valid. This is across any conditions. Reliability means that you'd get a similar score if you took it again. Validity means that the test actually measures what we think it does, right? So if it's a memory test, it has to actually correlate with memory abilities, right? Ecological validity is, does the test result reflect how you function in daily life? So does it really capture that everyday experience? And sensitivity is sort of the test's ability to correctly identify people who have cognitive impairment. So that's like a true positive, whereas specificity is its ability to correctly identify people who don't have impairment. So a true negative. These are just some standards that we hold measurement tools to. But the catch here is that a test can have excellent reliability and construct validity, but have terrible ecological validity, right? So maybe it is actually measuring, true memory function, which we think of in the hippocampus, right? And maybe the test is reliable, meaning that if you take the same test over and over again, you're going to get exactly the same score. But maybe it doesn't reflect the everyday functioning that you're experiencing. And so it might be scientifically sound, but clinically useless is what that means for capturing your lived experience. And a test can also be highly sensitive, right? But very low specificity, which leads to false positives and risks over pathologizing. And conversely, can be highly specific, but not sensitive enough, which may not capture all of the more subtle cases. And then finally, the other factor to consider is that a lot of these traditional cognitive tests were designed to detect severe impairments, right? So things like dementia, traumatic brain injury, stroke, they're looking for big signals. But the cognitive changes in ME and long COVID, for example, they may be more subtle, number one. They often fluctuate, number two, and they're context dependent. I've gotten a lot of people who come in and say, like, I've been resting for 2 weeks just to come in so that I can do this test. And so, in those cases, we may not always capture exactly what the person is going through. So context really matters. Cognitive testing, when you come in, often happens in sort of a quiet room. It's free from distractions. You're motivated, you get breaks. But as we know, daily life is full of noise, interruptions, requires you to multitask constantly. You're constantly contending with different distractions. And so it also requires a sustained effort. So these are just important things to think about with respect to measurement. Um, and one other thing that I wanted to say about that actually is that I think, a test that has maybe low sensitivity might miss your impairment entirely and give you sort of that normal score, even though your cognition has changed dramatically. So I would say, like, it's really important to communicate with the neuropsychologist or whoever will be administering any kind of measures beforehand so that you can really make sure that you're sort of capturing your real world experience of these impairments. It's also a little bit of a trade-off with tests that can sometimes be more fatiguing than others. So for example, we have a test of sustained attention, which can take like 15 minutes to complete. And so some neuropsychologists may not want to give that out of fear of sort of, causing an exacerbation, which is completely understandable. The issue, though, is that sometimes those are the tests that really do capture your real world experience. And so it's a little bit of a trade-off, but it's really important to just have that informed consent from the provider. And if there are any providers here listening, that is one of the most important things, right? Is talking to the patients beforehand and letting them know what to expect and that it is a risk with any kind of cognitive testing that, could trigger a PEM episode. And then, so the main takeaways and sort of conclusions that I wanted to just drive home today is to first trust your experience, right? If you know that your cognition has changed, that knowledge is valid. And even if tests aren't capturing it, you are the expert in your own functioning and you need to advocate for yourself. And advocate not just for, getting an assessment, but getting a comprehensive assessment. So if you're being evaluated, it's important to ask whether the testing will include measures of sustained effort, as I mentioned, assessment on different days, maybe to capture fluctuations, and patient-reported outcomes as well. So like the subjective tests that we were talking about, not just our traditional objective tests. And important to also talk to the clinician beforehand about whether you should be resting for 2 weeks or whether you should just be in your usual state when you come in. Third is just, document your experience. It's important to sort of keep notes about your cognitive difficulties in real world context. That really helps us sort of formulate and think through what might be going on before you even sit down to take a test. This information is actually really valuable for clinicians and for researchers, even if it's not a test score per se. Fourth is, understand that, again, normal results don't invalidate your experience. They reflect the limitations of our tools, not the absence of real impairment. And for, neuropsychologists who are savvy with infection-associated chronic conditions and who are aware of these things, they will interpret it in the context of your premorbid abilities and in the context of normative data as well. And then finally, just, again, be cautious about overexertion during testing. If cognitive testing does trigger a crash, that's important information, and you need to make sure that your clinician is aware of that. And so thank you so much for your time, everybody. And I'm including my email here in case anybody has any questions or concerns. I'm happy to chat further.
Awesome. Thank you so much. That was such a great presentation. And I think we need that to kind of understand and advocate for ourselves. But also, I think it's going to be helpful as we think about designing better research, because I think we have to be able to also talk about how we're going to do that job well in research. Because right now, we're not always getting the results that we expect we should get. So that's part of it.
Absolutely. Yeah, and that's why, we've been working together to think through this, because especially in randomized clinical trials and when we're evaluating treatments, we need to be thoughtful about sort of the baseline measures and how we're going to measure change over time, and actually capture those changes in the real world. So,
Absolutely. Well, thank you. And you'll be back for the panel.
yes, I'll be back for the panel.
Awesome.
Thank you
And there
so
might even
much.
be a couple questions for you, so feel free to go in there and answer them.
I will do that. Thank
Okay,
you.
thanks, Jackie.
Thank you.
Alain,
I'm here.
our very own Alain Moreau is up next. And he's
Thank
going
you.
to give us a presentation on, we have precision medicine for ME.
So you should start to see my screen now. So again, thank you very much for your great job as organizer and moderator, Sabrina. You're really an icon for this network and all the members and patient partners that are supporting us.
Oh my goodness, I'm so glad we got that recorded. No, I'm just kidding.
So I would like to acknowledge, starting with all the participants. So I came in this field 10 years ago now as a newcomer, and you received me with a very warm welcome. And we have tested so far over 600 participants. And I had the opportunity to work with many patient partners that I use the purple minion to illustrate them. If you are not familiar with my presentation style, I got the inspiration from patient partners, because often you are using purple minions to illustrate your health condition, and by respect, and you gave me the inspiration, I kept the minion and as often as a way to illustrate some stories about research in the field of ME/CFS. I would like also to acknowledge my team member, and a special thanks to our ME Stars of Tomorrow that you encountered yesterday. So many thanks for your feedback and great comments and questions for them, as well as I would like to introduce and thanks my international collaborators that are interacting with us on a daily basis on different projects and sub-projects. So here are my disclosures. So where are the source of my funding for different research program and also other volunteer activities that I'm doing on different boards and foundations. So as of now, we know that there is no pharmacological or interventional treatment for ME that has really demonstrated unequivocal, reproducible therapeutic benefits in a large-scale, well-controlled clinical trial. Even the most promising intervention have yielded mixed or modest results, often undermined by study design and limitation. It doesn't mean that it's not working. It just means that there are a lot of challenges ahead of us. So, as I said, many clinical trials have failed, not necessarily because the interventions were ineffective, but because of poor stratification, working with heterogeneous population, and a lack of mechanistic understanding. So often, some investigators try drug A for a treatment because they know what the drug A is supposed to do, but sometimes the effects are more like off-target effect, which are real effect, but way different than what you expect. So that may explain why a subgroup of your participants can respond. So that's why I proposed a few years ago at the NIH meeting, we should do a post-hoc molecular autopsy approach to reveal hidden responder subgroup. Even when a clinical trial failed, we may learn in much more details about how the drug A, B, or C works, and what we can learn, and maybe we may discover that some subgroup can respond. So, and this is part of the complex multi-system nature of ME. So failure is also part of a learning process in science and medicine. So we should open to such challenges. Of course, there are challenges that predispose clinical trial failure. Need to be naive to any prior treatment. Very difficult with ME patients because you guys are testing different supplements, having different drugs to attenuate many of your symptoms. And it's very difficult to integrate you to a new trial even if we do some week or 2 weeks of washout effect, some drugs must remain in place because you have severe cardiovascular conditions or other treatments. And being really naive to treatment, I don't think it's a realistic situation. So we need to develop more pragmatic thinking in the way we design clinical treatment. Slow ramping of the drug, whatever the drug we want to test, is very important. And often we shouldn't assume in a clinical trial for patients with ME the same thinking than a regular clinical trial because you guys have difficulties or tolerance toward different drugs. So we need to learn from those processes. Okay, it's well known for us from the field of ME, but not necessarily from clinical trialists that are used to go on different trials and administer different treatments. So slow ramping, meaning be cautious, but also we need to be more targeted to increase efficacy, especially toward low dosing. And that will cost more money because that may take more time. Lack of patient selection. There are some power issue here. So if we do a better mechanistic understanding, we may select only of those that we are thinking that are best responder and instead to blindly test the whole ME population. Tendency to reduce trial duration for economic issues. So also we need to be patient. And of course, lack of monitoring tools that may be a game changer in the field to really assess whether the treatment is working and how it's working. So having this type of molecular autopsy approach is maybe an option that is rarely pursued because there's a risk of flawed assumption or post-study design, and sponsor and investigator don't want to be exposed and often prefer to avoid that, which is, in my opinion, a big mistake. We can learn a lot working together, and we may, in some situation, even salvage a clinical trial that failed. So why precision medicine matter here and what we need for future clinical trial? So of course, we need to have a biomarker-based stratification to cluster patients by either immunological, metabolic, or neurological profile to test precision therapies. We need to pay attention of the difference between male and female because some key biomarker may be very different, and this is well known in medicine. We also need to develop objective real-world outcome measure, okay, heart rate variation using connected vest, for instance, measuring actigraphy to make sure how many steps are you doing in a day at home to follow you from baseline to a different time point in the clinical trial. There's also cognitive testing and brain imaging, okay, to track meaningful changes to really have more objective outcome measure and not just say to fill a questionnaire and say, oh, are you feeling better? Yes. So we don't want this. We really want to make a difference and collect with the participants real objective measures involving large multi-center, double-blind, randomized clinical trial. Better to start slow with one single center, but eventually would be also important to move toward multi-center. ME is not a rare disease, so it's not a difficulty to recruit patients technically. So, and trials should be adequately powered and conducted across multiple centers to confirm reproducibility. It's always good to work in the hand of the investigator in charge or the sponsor, but it must be also replicated by other centers, by other physicians as well. We need to have a better design to decentralize trials to involve severe and bedbound patients, okay? So those that are housebound or bedbound, to having home-based protocol to capture the full disease spectrum. I think it's possible and doable and must be done. We also, and we mentioned that before, to include also pediatric population, not just adult. We need to capture longitudinal study design to capture delay or sustained response and fluctuation of symptom over time. We need also to address whether there's a big difference if we give the drug in the morning or late at night, or at least at the beginning of the evening, to either reduce side effect or to increase the potential therapeutic effect. There are also some seasonality of the biomarker we are using, and that may apply in the way that we are responding to drugs. I put a cartoon also to introduce the concept of the altitude. So if you are at Salt Lake City at 3,000 feet of altitude, you may have a different or slight different physiology than people in New York City at sea level. So that will change your biomarkers and maybe the response to the drug A, B, or C over time. I know Dr. Petrino will talk about adaptive trials, so I use the octopus to symbolize adaptive trial, which may be as solutions to test multiple drugs in the same cohort of participants to save time and further introduce new knowledge with at the same time that we can test new drugs. So right now, the current strategy is to enroll a group of participants with ME, with a clinical diagnosis with different type of inclusion like the Canadian consensus criteria, and to exclude them for many reasons. So again, I strongly push for pragmatic essay. In the real life, our adults with ME have different comorbid conditions, and we need to take care of those and not just to exclude them, to have some kind of clean slate that nobody can reproduce. So, and the new strategy would probably benefit of the actual ME cohort that myself and many others develop over time, where we have well-characterized, deep phenotyped patients that we can make available for a clinical trial, and we can even pre-select individual based on mechanistic understanding of the drug that can be the good or the best responder, and to save money and increase the efficacy to develop drugs in clinics and put it in the market. What do we want to treat? Fatigue, dysautonomia, PEM, of course, PEM, as Todd mentioned, brain fog, sleep disturbances, all of them are important, but I think one of the very important things is the crashes. Patient partner told me many times that if we can eliminate all those crashes often relate to PEM, they will have almost a normal life, even if other factors may remain present. So we need to listen to the patient. So when I say we, is the community. So this is my warning slide, because the last 3 slides will be a bit more clinically oriented with some issues. Okay, so it's important to pay attention of the warning side, because the tests as well have some therapeutic options are not approved yet by regulatory agencies. So we developed a blood test called PredictMe that we published in 2020 and a new version in 2023 that can, with a very, very high accuracy involving a provocation maneuver inducing PEM with a cough, that we can learn who is ME plus FM, and who is FM. In fact, the test is very accurate for FM and ME-FM, which are almost 100% accurate to predict them. The tests also have the opportunity, using a panel of circulating microRNA, to predict subgroup of ME patients associated with specific symptoms like increase of orthostatic intolerance or brain fog in different subgroups that can speed up the process to discover relevant biomarkers as well as new therapeutic targets. So there is no comparable tests so far in the market or in the field of discovery. The test, and I give you an example on the right side of the screen, is also used to predict the therapeutic response. So I give often this example that if you have a high level of the EMIR 6819-3P, you will reduce TLR3, and if you reduce the TLR3, you won't respond to Ampligen, for instance. So the tests are not just predicting responder or non-responder to Ampligen, but it can predict many other therapeutic molecules. So again, more efficient, we can speed up the process or de-risk a clinical trial by integrating these information. We published recently a paper about sphingomyelinase called SMPDL3B, and we learned two things. First, this is one of the rare studies has been independently replicated, so in Canadian and Norwegians, and more recently in a UK cohort by Ametica Health, so which is a company based in UK that tests UK people and replicate our result by using RNA-seq by a different method. So they end up with the same response with an elevation of an enzyme called PEPLC, which cleave up SMPDL3B, which normally is anchored at the surface. So I will activate my pointer. So normally SMP is at the surface and is modulate immune response. And when it's cleaved up, the soluble form is associated. So more soluble form you have in your bloodstream, more severely you are affected. This is what this paper is telling us. Women are more affected because there is an interplay between estrogens and this enzyme and release more soluble SMPDL3B. What we also discover is you may have the same phenomenon, but because you produce not enough SMPDL3B. So the enzyme is not there. It's just that at the membrane level, you don't have enough production. And this occurs more often in male. So if you look at this paper in the supplementary file information, you will see the difference between male and females here. The good news is we find a drug called saxagliptin that is used in diabetes, well known by physician, and now we can combine it with myoinositol and for air to block this enzyme, the PACMAN, the PPLC. But if you don't have enough, it's not blocking the enzyme. Your issue is to produce more, and there is another diabetes drug, metformin, that can be given. The warning here is the fact that you cannot blindly use either of them, because if you are in that group and you take metformin, you will just increase your soluble SMPDL3B, and that would be detrimental to your health. So be aware of my warning and caution sign.
Alain, 1 more minute.
Thank you. So the other example is about FGF21, which is a fibroblast growth factor, which we present as a disease modifier because we have 3 conditions. You may have a very low level or very high and be between. So in between is not a matter of concern, but if you have very, very low, this is where that your PEM severity is very, very elevated. You have more autonomic immune symptom in the context of ME. In the FM, you have a high increased perceptual fatigue, which is probably through the centralization effect of FGF21. Elevation is not normal, but it may be, we suspect, and this is what we describe in this paper as a compensatory mechanism. And there are different strategies that we evolve and mention in the paper. And recently, we can remove the fibroblast activating protease where we did some measurement and it's not elevated, so there is no need to inhibit that enzyme. So metformin won't be a solution to increase FGF21, or at least its signaling effect, but others remain a good candidate. And there's a way, and there are different molecules that can reduce high circulating FGF21. Finally, we did a recent publication about an acute phase protein called aptaglobin, which detoxifies free hemoglobin in the blood. And we find that there is a genetic predisposition. So you are either aptaglobin 1,1, 2,1, or 2,2. 1,1, you are more resilient, but you have twice less aptaglobin 1,1 in the ME community, at least in our cohort in Quebec. And you have more 2,1, 2,2, and 2,1, you are more affected cognitively speaking and at the PAM level. So it's important because this is a genetic predisposition. There is nothing else you can do. But there's a way to attenuate those symptoms. So of course, there are treatment exists for some blood disorder where you release more hemoglobin. So there are intravenous recombinant on 1,1, which is non-realistic. We cannot treat all the 2,1 and 2,2 population by such intravenous recombinant, but there's a way to increase the deformability of red blood cells to prevent their release of hemoglobin. And again, those are suggestions. We need to set up a clinical trial to prove it, but the possibility exists and it's being tested for other conditions as well. So finally, EMI trial failed not necessarily from weak treatments, but from post-stratification and biological oversimplification. We can benefit to reanalyze past trial and integrate precision medicines to new design. And we need to power those future trials by biomarker guided and free from reliance on subjective endpoint. So thank you all for your time and attention.
Thank you so much, Alain. This was really great, and there's some questions, I think, and you'll be back to join us for the panel.
Yes, thank you.
Perfect. Dr. Petrino, I believe you're there. You can turn your camera on and get your slides started whenever you're ready. And Dr. Petrino is going to be talking to us about adaptive clinical trials and accessibility and research design, which we're thrilled about because we know you're doing some great stuff at your center, and we'd like to learn a little bit about how you're making research more accessible.
Thank you, Sabrina. Thank you so much for having me. Speaking of accessibility, this is my first time with the camera on, so I'm just going to turn it around so that we're not blinding half the audience. And I apologize, I'm sort of between conferences right now. I'm at a different conference at the moment, so this was the only room I could find. Okay, I'm going to go ahead and share my slides.
Thank you for making time for us.
Oh, of course. No, it's an honor to be here, and I just want to make sure that we can make this all work. Okay. All right. I'm thinking that's working. So yeah, I wanted to talk about
your slides have a couple of little boxes. Well, I think it might just be maybe
gone now.
I was going to say there's still one on the right-hand side.
Oh.
Over the logo.
I don't know what to do about that.
Okay.
I can't see it. Is it big?
It's blocking part of your bottom of your slide, like probably like an eighth of your slide or something. I'm not good at math right now. I'm too tired.
Oh no. Okay, let me just try and stop share and reshare and see if that fixes it.
Okay.
Sorry, I'm with a different computer, a different kind of everything. Is it gone now?
Nope. And there's two more in the center.
Oh no.
Oh, the center one's disappeared. So it's the one on the side. Maybe if you go to the next slide, maybe it's not going to be there. I think it might be your internet because it seems to be jumping all over the screen.
Oh dear. Is it gone on the next slide?
No, it's not. I don't know that we have a copy of your slides either. If you wanted to send a copy.
We are going to try one more thing. Let's just
We'd be happy to flip them for you if you need us to, but I don't think we have a copy.
I sent you a copy this morning and then I received an email saying it bounced back because it was too big. I'm so sorry. Let me do it this way. One moment. I'm going to go ahead and...
This just gives the attendees a little peek into the back scene of what goes on to get all this up and running. It's good.
Okay. Let's see. Okay, let's see if we can do it this way. And...
Well, I see two of you. Oh, now I see you and I see the slides. But they're really not going to be big enough for us to be able to read much on them, probably.
Let's try one more thing. How about that?
Oh, I see your slides really well right now. Can't promise anything for the future, but it's looking good.
All right. It's okay. We've overcome. All right. So it's an absolute pleasure to be here. Thank you for bearing with me as we figured that out with a brand new laptop. And I wanted to talk a little bit about some of the complexity we face with clinical trials and in ME and other infection-associated chronic conditions and illnesses, and some of the ways that we have been overcoming these issues, as well as some of the ways that we're, thinking about clinical trial design for the future. To start off, obviously, we call them complex chronic illnesses for a reason. They are complex, and don't worry if you can't see all the words on this slide. You're not supposed to. You're just supposed to take away the message that, with a single diagnosis like long COVID or ME, you can have a whole lot of things going on and a whole lot of clinical complexity with your presentation. And one of the things that we have tried to do with our own sort of clinical guide for infection-associated chronic conditions and illnesses is build clinical guidelines and clinical suggestions for how to embrace this complexity in a clinical setting. But that doesn't solve the problem of how do we raise the bar of research so that we can go from having just, a handful of clinicians who feel confident embracing this level of complexity with off-label use of medications and all of the challenges, complexity, and expense that comes with that, to actually having FDA-approved medications, actually having drugs that are on label for specific diagnoses. And how do we start to untangle that? And I'm going to start off by saying that our status quo of research right now, we have a huge problem. We're not, in my opinion, taking adequate steps to address complexity. And we're jumping into large-scale monotherapeutic trials, saying, we're just going to throw everything, everything at this one drug and hope for a silver bullet. And in a population that is as diverse as ME, not only is this probably not going to work, but it may in fact have negative consequences on engaging pharma and getting medications labeled for specific uses, right down to the fact that you may end up with people saying, well, they tried the IVIG trial and it didn't really work. So, I don't think that you should try IVIG because there's a paper saying it doesn't quite work. And so I think that we really need to rethink the way that we're doing trials. We really need to rethink the way that we're publishing papers and talking about the data that we're collecting so that we can avoid these issues. And, and unfortunately, that's probably going to be a little uncomfortable. I think we need some radical change in the way that we do clinical trials. And what I wanted to do was sort of break down some of the needs that I see emerging in the community. This is me speaking as someone who has been a driver for innovation in multiple fields over the last 2 decades in my research career, really thinking about when a field is stuck, how do we pivot? How do we try to do things differently so that we can make real progress? So the first thing that I think that we need is the ability to perform advanced responder analysis in every single trial that is conducted. You don't need a massive trial to do adequate responder analysis. You just need to be thoughtful about the way that you're conducting the trial, such that if somebody does respond, you can understand who they are and why they may be responded. And a lot of the work that we do at the Cohen Center for Recovery from Complex Chronic Illness, which is a hybrid clinical and research center for infection-associated chronic conditions and illnesses, is we will run these rapid interventional trials. So we won't go for this huge N, 1,000 people on GLP-1s. I don't know why that one came into my mind, but we won't run these huge trials. We will run smaller trials, but at every point of assessment, we will do these deep phenotyping techniques. We work with wonderful people like Akiko Iwasaki at Yale University. We work with Polybio. We work with the J. Craig Venner Institute, so that we can, with every sample that we collect, we can be doing deep phenotyping techniques so that we can really understand who is in our trials, what is happening from a molecular level all the way through to the physiological level of people who are sitting in front of us. What that allows us to do is at the end of the trial, when inevitably, and I say inevitably, with certainty, not every single person in the trial responds, it allows us to understand, okay, actually we had a 20% responder rate in this trial. Like 20% of people actually did really, really well. Was that just a placebo? Was that just a coincidence? Or can we actually see something about that 20% that was special? Were they testing positive for a specific enterovirus? Were they the folks who had neuroinflammation markers that were going off the charts? Were they the folks who had evidence of mitochondrial dysfunction? Who were the people who responded? Once we can identify with a biomarker who the people that responded are, that's when we can run the large trial. That's when we can get really specific about our inclusion criteria using the novel biomarker that we've identified. And that's when we can get one step closer to labeled uses of therapeutics and personalized medicine. That's why when we can say, hey, if you have an ME diagnosis and this biomarker, we can prescribe this drug and insurance will cover it. And I know that that's, a lofty goal. A lot of people might be sort of sitting back saying, we've heard all of this before, but, it doesn't really work out in practice. It does work out in practice. A concrete example of this is Øystein Flug's work with daratumumab, which is a B-cell therapy, which his team published just this year in 2025, where if you look at the people who responded to daratumumab, you'll see that there is this beautiful separation between responders and non-responders based on natural killer cell count. So the individuals who had a low, under 150 natural killer cell count did not respond at all to the drug, or in fact, in some cases they even had an adverse reaction. The drug didn't make them feel well. Whereas those who did respond had a natural killer cell count over 150. And what Flug's team have now done is they're going to run a new trial on daratumumab, but this time, they're only going to recruit folks with a natural killer cell count of over 150. Now, crucially, again, this discovery and this really interesting and exciting finding was made not with a 250-person clinical trial. It was made with 10 people, and it was made with really a lot of care taken to deeply phenotype these folks so that we could understand who might respond and who might not respond. So that's our first need. We need to be taking every opportunity that we can to be phenotyping these folks. Our next need is that we need to start developing and really thinking about methodologies that allow for combination therapies to be trialed. In the manual that I showed the QR code for at the beginning of this talk, we talk about some of the drivers of symptoms in infection-associated chronic conditions and illnesses. The reason we do this is because when we talk about a condition like ME, or a diagnosis like ME, I should say, there are many symptoms associated with ME. Just like long COVID, there can literally be hundreds of symptoms associated with ME. However, to me at least, and to the ME patients that come through our clinic, it doesn't make a whole lot of sense to say, what's the cure for fatigue in ME? What makes more sense is to say, why is someone with ME getting fatigue? Are they getting fatigue because they have mitochondrial dysfunction? Are they getting fatigue because they have reactivated EBV and other herpes viruses in their body? Are they getting fatigue because of autonomic nervous system dysfunction? What is driving the fatigue that they're experiencing? And then we can talk about treatments. And in our manual, we identified 10 key drivers because these were the drivers that we felt had the most evidence behind them, the most publications saying these things correlate with fatigue or post-exertional malaise or migraines or, any other sort of diagnosis or symptom associated with these IACCIs. Now, the other thing that is important to note is you may have more than one of these. You may have all of these, you may have all of these and more that we haven't yet named as key drivers, such as joint hypermobility spectrum disorders. So it's really important to understand that a silver bullet approach to trying to treat folks, again, is going to fail because you're not necessarily addressing the individual drivers that are affecting your patients. That's where methodologies like adaptive platform trials can really be helpful. These are expensive, these are involved, but they really do allow us, so long as you are very hypothesis driven in what you're doing, they really do allow us to test for multiple combination interventions. This is a nice little schematic showing how adaptive platform trials work. That was published back in 2020. These methodologies are not new. They are being used in other complex conditions, such as cancer research is leading the way, I feel, in adaptive platform trial methodology. And I think that, it really shows us that what you can do is create solid scientific methodology around non-traditional clinical trial infrastructure that allows us to be highly dynamic and as complex as the disease process that we're trying to cure in our research methodology. And so, the way that an adaptive platform trial works is you may start out with 4 or 5 arms of a single intervention. Or you may start out with 4 or 5 arms of different combinations of interventions. And then as a cohort of patients goes through that first 4 or 5 arms, you stop. You do some data analysis, or, in the case of pure adaptive platform trials, an algorithm does the data analysis for you in a very unbiased way and says, you know what, drop that arm, drop arm number 2, take intervention 1 from arm 2 and add it to arm number 1 as another intervention and start all over again. And then reanalyze as you go through another cohort. Figure out what's working, start all over again, and so on and so on and so on until you have a combination of interventions that is working for each subset of the patients that you have. This sort of dynamic and living clinical trial design is really, in my opinion, something that is crucially missing from our attempts to address infection-associated chronic conditions and illnesses. And we have the blueprint. We know how to do this. So it really is just a matter of convincing funding agencies, pharma companies, et cetera, to collaborate in public-private partnerships on creating these sorts of interventional trial infrastructures. Moving into our third need, we need to rethink accessibility of mainstream clinical trial design. I was already, this is already, part of my presentation, obviously, but I just want to name the fact that, sorry, Sabrina, I've been creeping on you on social media. I see that you're tired. I see that you're tired running a virtual conference. I think that this is just a huge part of accessibility in clinical trials. Clinical trials ask a lot of people with ME and with other energy-limiting complex chronic illnesses. And when we think about some of the most recent literature on ME, we know that roughly a quarter of folks with ME are either homebound or bedbound. And more, a large proportion more of people with ME, they can maybe leave the house, but they're going to pay for it afterwards. And they can maybe go to work, but they don't have much left in their energy envelope to go to a clinic and engage in a clinical trial. So the idea that we're running clinical trials that require location-based intervention is a real challenge when a primary symptom of ME is post-exertional malaise. And this is not you know, just again, this is not, I'm not here to just talk about aspirational things. You know, these are concrete things that need to be considered and taken seriously by regulatory agencies, by government research, you know, by government entities that sponsor research, because it's a fundamental human right. You know, this is from the United Nations Article 3 from their, you know, a gathering in 2006, saying that all folks with a disability have a fundamental right to inherent dignity, individual autonomy, and full and effective participation and inclusion in society. When we exclude bed and homebound people from clinical trials, we're not meeting these fundamental human rights. So at every given opportunity, we need to be thinking about decentralization. We need to be thinking about engaging home and bedbound folks into what we're doing. And, you know, I will be the first to raise my hand and say, we are failing at that sometimes in our clinical trials. We do have fully decentralized clinical trials that we run, but sometimes, you know, there are reasons why we require a visit of our participants. And that makes, you know, to my next point, further innovation is required in the way that we measure a lot of the primary endpoints in our clinical trials. I think that we need to start thinking about jettisoning old school, old-fashioned technologies and clinical endpoints. I do not think that clinical trial endpoints that involve exercising patients performing CPET are appropriate moving forwards. And I know that this may be controversial for some, but I think that we need to move beyond it. I am not discounting the importance of these methods in giving us insights into physiology, but I don't think that they're appropriate as endpoints anymore. I think we need to move forward, and I think we need to do better. I think we need to talk about the fact that we have multi-omic analyses. We can do deep immune profiling on patients. We can do proteomics. We can do transcriptomics to understand what is happening on a pathogenic, you know, a persistent pathogen level, what is happening in terms of your body's clotting factors and your endothelial health, et cetera. We have advanced physiologic monitoring technologies. So we have, you know, technologies like Mira, Femtech, that allow us to do daily analysis of hormones of ovulation. We have continuous glucose monitoring. There's also now wearables that do continuous cytokine monitoring from sweat analytics. We can do continuous cortisol monitoring. We can do continuous blood lactate monitoring. These are all things that can give us insights into how hard we're pushing a person that without actually exercising them and putting them on a treadmill and doing a CPET on them. That needs to go. We also have enhanced tissue analytics. We've got, you know, if you tune into, you know, the Polybio Symposium next Friday, you're going to see all of these incredible teams doing outstanding work that looks into how to analyze tissue in a way that provides insights. If you look at what's pictured here, this is work from Rob Wust showing that folks with ME and long COVID, so pre-2020 ME and long COVID, have very concrete changes in their capillary structure that correlates to post-exertional malaise. So these are things that can be done. We're also working with a company right now that is able to provide VO2 max from proteomics that's taken from blood. So we're able to move beyond having to put someone through a potentially, you know, a strenuous and potentially harmful test in order to gather information about their exercise tolerance, which, you know, I just think that we need to move beyond. And you know, finally, we have platforms like Visible that allow us to do continuous and remote digital monitoring. So rather than doing a questionnaire once every 3 months to see how someone's doing in a clinical trial, we can be asking them hourly. We can be asking them daily. We can be asking them weekly, and you get the point. Again, this is not a fantasy. You know, we've got concrete examples of this. We completed a decentralized clinical trial in long COVID not so long ago. You know, we published it earlier this year. And what was, you know, interesting about this was we recruited patients from every single contiguous state in the United States. Everyone was recruited from home. All study activities happened from home. And we were able to recruit 4 to 6 times faster than traditional centralized trials that required people to show up in person. So not only do these decentralized trials work faster, but of course you get a more diverse population, because you're not just relying on folks who are close to academic medical centers. So you learn more about the illness as well. And if you've got someone who's homebound or bedbound, who we can learn so much from, because their symptoms are much more severe, but we're often not learning about, their symptoms directly, because, they're frankly invisible and missing from our current research infrastructure. Decentralized clinical trial design helps us get past that. So my final comment, my final need, which I'm not going to go into multiple slides on this. I think that in addition to everything I've just said, there is a fundamental need for more organization, more community, more infrastructure in the research that we're performing. What that looks like is, of course, at all times, we need community engagement. I need to know from the community what's working so I can run a clinical trial on it. There is so much wisdom, not just in communities of patients. Of course, there is a ton of wisdom in communities of patients, as we know, but also in communities of these doctors who are figuring it out, without accolade, without any sort of fanfare, in the middle of nowhere, who also have great ideas and insights that we need to be mining and using resources to run good clinical trials on. I can't get it out of my mind how fortunate I am to have met Dr. Skip Pridgeon in Alabama, who has been using combination antivirals in his long COVID patients to outstanding results. And he was just doing that in the background, on his own. And it's only because we were having conversations with patients who said, oh yeah, I went to Alabama to get this treatment because I heard about it from another patient who heard about it from another patient. So we can never stop mining communities to understand word on the street. What's the most exciting thing happening? What should we be trialing? Help us to steer in clinical trial targets. We need organization. That means clinical trial networks. That means repositories. So all of those tissue and blood samples that I'm talking about, bank them. Take a little extra vial so that if somebody else comes to you 5 years later and says, I know that trial of yours failed, but I saw something interesting in the data. Do you have any blood left over? You can say yes. It doesn't take a whole lot of money to take an extra vial of blood. So bank extra samples. Don't be wasteful of tissue and samples that people so freely give in clinical trials, because it could be, the next breakthrough that happens. And so our final need here is to really bring a lot of organization, community, and infrastructure to the trial process, whether you're doing a tiny little exploratory first responder trial all the way through to 1,000-person double-blind, placebo-controlled RCT or adaptive platform trial. So my final 2 points are, if there are some researchers and clinicians watching this who are very comfortable in the status quo, saying, well, I'm just going to keep doing things the way that, I'm going to keep doing them. And this whole newfangled way of thinking about things, I'm not so sure about. I'm going to just repeat what a patient told me just yesterday, actually. If you can believe you can do it, or you believe you can't do it, you're right. So every innovation that we have led in our team's history of working in innovation has really focused on this idea of we believe we can do it. And so we're going to go out there and do it. And that's what we owe our patients. That's the energy that we owe our patients to bring to the table. So that's what I would say to my colleagues. And, and to really double down on that in my final thoughts, I will say right now that we're at a very exciting point in ME research. I think we understand this illness better than we ever have. And we need to meet the moment with innovation, with passion, and with increased pace and purpose. Because if we do what we've always done, we'll get what we always got, which is failed clinical trials and no labeled drugs, no FDA-approved drugs for a patient population that deserves better. So I will end it there. I'll thank you, everyone, for putting up with all my tech issues, and just acknowledge all the wonderful people who support me.
Thank you so much. That was so much great information. And, you know, we have a lot to learn, I think, in terms of how we move forward. And I think everybody here today is agreeing that there's definitely an opportunity here for change and for growth and for innovation and for increased accessibility. So thank you so much for drawing some parallels to some of that and showing us some of what you're working on. You're not going to be with us for the panel today, but maybe we could have you back for a webinar sometime.
Yeah, that would be great. Thank you so
Okay,
much.
wonderful. Talk soon. And Michael Van Elzecker is up next. And Michael, am I saying your name correctly?
Yeah, that's perfect.
Okay, been a big fan for a long time, and I just thought the other day, I hope I haven't been saying his name wrong that whole time. So you're going to talk to us about a topic that is incredibly important to this community, and we want more research in, and we want more understanding in. We can't actually see your whole slide yet.
Almost. How about now?
It's perfect.
Okay, great.
And just a reminder for everybody listening that we're not doing bios just because everybody is so brilliant. It would take forever. So please go to your conference programs and you'll see all the bios there so that you can know who these individuals are a little bit more deeply. And you can get started whenever you're ready.
Great. Thank you so much. So yeah, I appreciate you guys having me here. I'm going to talk a little bit about neuroinflammation as a general topic and how it may relate to cognitive and some neuroautonomic dysfunction that we see in ME. And I want to just give a disclaimer up front that you know, we have been trying to study ME for a long time. You know, Amy Proal and I formed a nonprofit so that we could study, you know, these patients that were neglected. And it formed in 2018, and then COVID hit, and we were able to get a pretty big chunk of funding. And so a lot of what we've done is to take long COVID research that we had funded and apply it to people that were sick before 2018. So I'm going to jump back and forth a little bit between people that got sick with COVID and people that got sick with ME. And just to, you know, reiterate what people before me have said, you know, with the ME diagnostic label, I don't think everybody has the exact same issue. It's possible and likely maybe that if we went back to study the outbreaks properly, that we could find a single thing. But the way that diagnostic label has been used, there's probably a diversity of presentations and even drivers that we have to you know, be aware of. So as everybody knows, when COVID hit, a lot of the media was kind of surprised that a certain subset of people weren't getting better. And unfortunately, I think a lot of us on this call were not surprised. You know, Amy, Dr. Proal and I got together and wrote a paper trying to guide the research of this, you know, new pathogen so that it would be properly studied and, you know, the drivers given proper weight, trying to deflect, you know, the idea that this was all a big mystery that we couldn't figure out and actually get into it. So a lot of what I'm going to talk about today is based in this paper that we wrote a couple of years ago about neuroinflammation, how to properly study it in the ME diagnosis and then in later conditions. So we have a lot going on in my lab right now. I'm just going to kind of advance these. Lots of different projects, and almost everything we're applying to people that got sick before 2019. Everything except for that last item actually is applied to both groups. So I think that's really useful because we can weigh in on mechanisms that are similar or different. Given that some of the symptoms are the same, that is an important question. And then what are the drivers that are different is an important question as well. So just to start with, I'm going to talk a little bit about this paper that we put out last year, where this is a study of long COVID patients that got sick, mostly early pandemic, you know, before Omicron. And what we did in this study, in order to try and get it out quickly, is we used controls, healthy controls that had been scanned before they ever got sick with COVID. And so what we're doing is following up with controls that recovered from COVID, as well as with people that got sick before 2019. So neuroinflammation, what we do is we use a type of a brain scanner called a PET scanner, and that measures by using a radioactively tagged chemical that binds to a particular protein in immune cells. And so microglia are the resident macrophage immune cell of the brain. So that is one of the more common types of brain cells, in addition to neurons, are glia, and especially microglia. So when microglia come in contact with either a pathogen or an antigen from a pathogen, or cytokines, things that might excite immune cells, they activate, which means that they kind of change shape, they withdraw, and they get sort of this thick phenotype. And what they do is to pump out both immune signaling, like cytokines, which are sort of immune molecules. They pump out things that excite neurons, like glutamate, lots of things, ATP, bradykinins, and as well as the TSPO, translocator protein. So the translocator protein is the thing that we actually measure in these PET scans. And that's what the PET scans can pick up. And we think that this neuroexcitation and immune signaling may be part of the reason that people, sometimes have a little bit of fuzzy thinking when they have this problem. So in this initial study, it was, mostly people that were not hospitalized with COVID, so they were a little less severe. And we used sort of a standard PET scan protocol. And again, we did this in people with ME as well, and that I'll talk about in a second, and people that had COVID and then never fully felt recovered, as well as people that had COVID and did feel recovered. So this is just some sort of stats information about what we did. This is the basic finding from this first pass analysis where we compared to COVID naive controls. And this is a heat map. So you're looking at statistics, basically. These are areas where there was a significant difference in the COVID cases versus the controls. We had mostly women in the long COVID group, and we did some careful things like, split out and did a woman-only analysis to make sure that wasn't driving the effect, et cetera. And it wasn't. So this is kind of the whole brain signal where each dot is an individual. I think it's important to note that the healthy controls, it's not just a lack of inflammation. In fact, we had somebody way up top in the healthy control group that seemed to have a lot of this signal. But you see the group statistics that show a pretty solid group difference. And I think that's important to keep in mind, that you can't just look at a scan and say, there it is, there's the illness. But these are some of the regions, the brain regions that came out significant. And, a lot of what we found was that these are regions where there are normally in everyone, sort of windows in the blood-brain barrier where larger molecules can kind of get through. And we just wanted to make sure that it wasn't driven by one brain region. So we kind of split into several different brain regions, and we found that the significant brain regions really did show a pattern where, again, the healthy controls had a spectrum, and so did the long COVID patients, but the spectrum was higher in the long COVID patients. This is an example brain region that showed increased activation. It's called the subfornical organ, and it's number 3 here on this image. And these are what are called circumventricular organs, which means that there's a window in the blood-brain barrier. So we thought that maybe there might be something in the blood that might be, predicting the inflammation in the brain, signaling across the blood-brain barrier. So we took blood, saliva, lots of other measures before anyone got into the scanner. And we found a couple of things that lined up, correlated pretty well with that neuroinflammation signaling. So, for example, over here on the left is called it's called L-selectin, which is what's called an adhesion molecule, which means what it does is it expresses on vasculature, and it causes circulating immune cells to attach and then get pulled into the tissue. So that includes neutrophils, which are an immune cell that I'll talk about in a second. And so we thought maybe that's important, and I'll get back to that in a second. And here's fibrinogen, just a sort of a clotting molecule that also correlated pretty well. And we found 6 total markers that predicted the neuroinflammation pretty well in those long COVID patients. So now we have another analysis where we have COVID recovered controls. And I just want to show the pattern here. And, pretty similar, although the pattern did kind of move into brainstem, which is one of the regions that we've been interested in a long time. And again, you can see the split between the cases and controls. It's not the case that everybody is higher than the controls, but they are largely as a group higher than the controls. And so here's some data that I haven't shown anywhere yet. We just pulled this together recently, showing some patients that have ME. And by the way, sorry that I didn't mention this. The long COVID patients all qualified for the ICC criteria of ME. That's not to say I think they have ME now or they converted or something like that. I don't really think of it that way. But they qualify for the ICC diagnosis, meaning that they have a lot of the same symptoms. And I think part of what we're getting at is, why do they have similar symptoms? What does that mean? These are ME patients that got sick before 2019. And in this case, it's 24 patients over 14 controls. And we do find significant neuroinflammation across a couple of different brain regions in the ME patients. This is a brain region called the anterior midcingulate. And I will come back to that in a moment. This is brainstem in the ME patients versus the COVID recovered controls that are healthy. And, you see a pretty good cluster of neuroinflammation in the ME patients. This is similar to what Nakatomi reported many years ago using sort of older methods, but it is not similar to what Raymakers reported in 2021, where they studied specifically a group of women that had been sick with Q fever and had been sick for a long time. So part of what we want to do is to really dig in and find out, what does length of illness have to say about this, sex, other measures in the blood and things like that. So we're interested in how this may affect cognition. We know that a lot of patients report that they have sort of trouble thinking, remembering, that they kind of get a brain fog if they have too much input over a period of time. And so what we try to do is to study something called top-down cognitive control. And that's kind of a fundamental aspect of cognition. And if you think about, if you're looking through a crowd for a friend with a red shirt, what you're doing is searching for something on purpose, right? And then let's say that all of a sudden, a red balloon floats up from the crowd. You're not looking for that, but it grabs your attention, even though you're looking for something else. That will mean that you cannot focus as well on the thing you wanted to focus on, and instead something grabs your attention. Well, this is a fundamental aspect of cognition that reflects a lot of different things, like even searching for a memory within your own mind. And it happens to be driven by that region that I talked about before called the anterior midcingulate. Well, it turns out that if you look at other literature, this anterior midcingulate structure is affected by inflammation. So this is a meta-analysis from a couple years ago, 2018, where they just took a bunch of studies of inflammation and brain imaging and looked and saw, what is the sickness circuitry? What are the areas that are affected by inflammation per se? And one of those regions is the area that we see a little bit more active in the ME patients versus controls. Well, interestingly, my PhD mentor, Lisa Shin, developed a task that really probes this brain region called the multi-source interference task that we call MSIT. And we thought that we have this task that can measure this brain region that's important in cognition. It's sensitive to inflammation. Well, let's check it out. So what we do is to put them in the task, put them in the scanner, and then we give them the task. And the task is to pick the numeral that is different than the other numerals on this button box. So see this button box right here? Picture your fingers, 1, 2, 3, index, middle, ring, and they're pressing the button. So the correct answer here is 1, and then 2, and then 3. The correct answer here is 3. Now that's hard because it doesn't line up with your finger that's on the 3 position, right? So that's what you kind of have to override. So the correct answer here is 2. The correct answer here is 1. So again, 2, 1, 3, that's pretty easy. 2, 3, 1, that's hard. And so when we give it to patients with long COVID, and this is an analysis that shows that they kind of need more real estate. It takes more effort for them to do this task. as you can see over here, this is fMRI data. So this is MRI data, and what we've got is a scanner that can do both at the same time, both kinds of neuroimaging at the same time. So what we did was to take the ME PET data, which is neuroinflammation, and we show that region right here. And then we overlay the task data. And if you can see right here, the blue is the task data. And what you see is it overlaps pretty well. And this is just a little pilot analysis in 9 patients versus controls. We have that more fleshed out now. And we think that that may be part of the reason that a lot of patients have trouble concentrating, is that's a brain region that's affected by inflammation. Interestingly, this is another form of data that we're doing with EEG. Now, forgive me, this is combined ME and long COVID. We just did this to get the power. We're going to do it separately. I don't, again, I don't think it's the same exact thing. But this is showing that the front and the back of the brain are desynchronized. They ought to be communicating really well when they do a task like this, and they don't seem to be doing that. And the task performance on that task anti-correlated with that what we call phase coupling in those in the patient group versus the controls. We have more patients now that have undergone that paradigm, and we'll split the 2 groups and take a look more. And tell me if I'm getting close to time here. So another brain region that we're interested in is the brainstem. These are 2 papers that I've written over the last couple of years, just arguing that brainstem is kind of a fundamental, important area to study. And it turns out from that same meta-analysis over here on the left, well, they're showing brainstem is really activated by peripheral inflammation. And it happens to be where the vagus nerve enters the brain from the body. The vagus nerve is something that detects inflammation in the periphery and sends a signal to the brain saying, hey, the body is sick, and maybe you shouldn't spend your time doing other things. You should just rest. So we did an analysis where we extracted chemical data from patients in the scanner, again, in that same scan session that we're doing everything else except for that EEG, the same scanner that we do the neuroinflammation and the fMRI MCIT task from this region of brainstem. And we see that in both long COVID and in the ME patients, we have reduced levels of what's called N-acetyl aspartate, which is kind of a measure of brain health. And again, it's not the case that I can just look at the scan and say, that's a patient, that's a control. But at the group level, it's more likely that the patients will have reduction in this chemical that's kind of associated with brain health. So I think that's sort of another way that we can get at it, and another sort of mechanism of problem. So before they get into the scanner, we do this kind of neat thing where we take blood from the patients and we don't freeze it. We take it right over to our collaborators across the street. And while the neutrophils, which are the form of immune cell that I talked about earlier, that kind of connect to vasculature, while they're still alive, we put them on a little plate where there's these little wells. So the circle parts here, inside of the circle parts, we put bacteria that the neutrophils, which are in white, swim in and go get, right? Which is their job. So we end up quantifying what we call chemotaxis, which is the ability for these cells to move along an axis and go into the little well and find the bacteria. Well, what we found was over here, long COVID, and in the middle, ME/CFS, again, sick before 2019, show more of this chemotaxis. The long COVID is just a trend level, but the ME/CFS showed enhanced chemotaxis relative to healthy controls. We think that what that means is that the immune system is primed. I know that's kind of like a duh thing, but what we can use is this assay to try and get at what exactly is doing it. What exactly are the antigens or perhaps priming that's driving that effect? And these are just the statistics. So another thing that we do before people get into the scanner is the 2 nights before the scan session, we give them an overnight wearable sleep device to measure their sleep quality. So it's this little device called Sleep Image, and it calculates a sleep quality index It combines stability, fragmentation, periodicity, and we see in that measure that the ME and the long COVID patients show worse sleep quality relative to healthy controls. This dashed line is what the company says should be sort of the normal healthy data. So again, the reason that we do this is now we can dig in and see, is there something that predicts this? Is there something we can measure and then perhaps try and address? So another thing, yet another thing that we do with our very generous patients that we do before the scanner is we do what's called pupillometry. So we put everybody into this darkened room. You know, photophobia is a big issue with a lot of patients. And we sort of deliberately flash a real quick light and watch what happens with their pupils. We get 2 measures per eye. So if you know statistics and you see the numbers, you're going to be like, did you have 500 patients? No, we just got 2 measures per eye. So 2 eyes per person. So a lot of measurements. So in this case, we do see differences between long COVID and ME/CFS. This is the initial pupil diameter where the long COVID patients as a group show a smaller pupil diameter, and then the long COVID patients as a group show also a reduced diameter after the contraction of that's caused by the light. So again, all the long COVID patients qualify for the ICC technically by just symptom. So I think that's kind of an interesting thing. We don't really know if it's length of illness that's driving this, but we'll dig in and figure that out. So here's an example where the ME patients are different than the controls. And in this case, it's the constriction velocity. How quick does the pupil snap shut? And this looks a little bit more like a concussion in the ME group, the pre-2019 group. This is the average velocity. And then this is max velocity, maximum velocity of that pupillary constriction in the ME group versus long COVID. And with that, I just want to say thank you. I'm sorry if I went a little bit over. I want to just thank my team that is, really good. Almost all the patients that go through our research program say that it was a really good experience, and that's really saying something because almost everybody has had bad experiences with healthcare, and we're not even trying to get people better. We're just trying to figure out what's wrong. In other words, we don't give anyone medicine. So the fact that everyone has a good experience is a real testament to the team, and thank you very much.
Thank you so much. That was fantastic. And a lot of great information that I think helps us to start to think a little bit more about how we can look at this type of research and fight for this type of research and where those innovations are. Are you going to be back for the panel? Oh, you're on
I'm going to
mute.
do my best. I technically have office hours, so it slightly depends if any students show up. I'm kind of cheating by joining, but I'm going to do my best to join at least for
Just
some of
lock
it.
the door. Just lock the
Yeah,
door.
yeah, exactly.
Okay, well, hopefully we'll see you back.
Thanks for putting this together, Sabrina. I know you worked really hard, so thank you. And the whole team, thank you.
Yes, thank you so much for that. We really appreciate it. We did work really hard, and while we still look like we're doing okay on camera, it's different. It's a different thing altogether behind the scenes. We're
Hang in
struggling.
there. You're doing great.
Thanks. Okay, guys, we have a break, and we're going to take it because rest is important. And so we're going to come back at nope, sorry, that's my time, 3:18 Eastern. We're going to be back. Hi, everybody. Welcome back. So we are going to get started with our next presentation, which is patient engagement in ME research with Carrie Anna McGinn and Trudy Flynn, two members of our planning team, two active members of our community leadership team as well on ICANN-CME, and two very passionate advocates in the community and patient partners, even more importantly. So they're going to talk a little to us about why this matters, why it's important, because we're trying to encourage more researchers to integrate this into their research, and we're trying to encourage more patients to want to get involved. So this is going to be a great presentation to get us there. So Trudy and Carrie, whenever you're ready, you guys can start.
Boy. 1 second. I want to go back to the beginning.
I have my video on. Oh, there I am. Okay.
There we go. No, I want to go back. Okay, sorry. All right, hello.
There you go.
Great. Hello, bonjour. Thank you for joining us today. Carrie and I will be speaking about patient engagement in research. So, having problems with my slide, especially in ME research. So, ideal patient engagement goes beyond simply including patients. It's about truly valuing their lived experience and perspective. Patient isn't just someone receiving care, but anyone with personal or caregiving experience of a condition that can inform better decisions and outcomes. When patients are engaged meaningfully in governance, setting priorities, research, and knowledge translation, there is a move from doing research for people to doing research with people. This collaboration ensures that what is studied and how it's shared reflects the needs and realities of the communities research aim to serve. It's important to understand the difference between patient engagement and research participants. Patient engagement means that patient partners are actively involved in shaping the research itself, using our lived experience to help set priorities, refine study designs, review materials, and share results. Our insights guide the process to make the research more relevant and meaningful. On the other hand, research participants are the people who take part in the research. For example, those with ME who complete surveys, interviews, or medical tests. Both roles are essential, but engagement ensures that research truly reflects the patient's voice from start to finish. The International Association of Public Participation, or the IAP2, outlines a spectrum of engagement from simply informing to fully empowering patients. While empowerment is the gold star, where patients' decisions are implemented, it is okay to aim for the collaborative level, which itself can be challenging to reach in today's research climate. At the informed stage, patients are only kept up to date. That's communication, that is not engagement. Consult involves asking for feedback through surveys or meetings, but still doesn't include patients in the decision-making, so that's not engagement either. Involve means working with patients to ensure their views are reflected, getting closer to patient engagement, but we're still not quite there. Collaborative is where true patient engagement happens. When patient partners share their lived experience and help shape decisions as equal team members, that is true patient engagement. Finally, the empower means patients either lead the research or make the final decisions, which is a level we continue to strive for in meaningful patient engagement. Over to you, Carrie.
Thanks, Trudy. All right, meaningful patient engagement can and should, when possible, take the form of patient partnership, where patient partners are considered equal partners at the table with shared power. Patient partners should be at the table when important decisions are made, such as research priorities and the questions that are being decided, as well as embedded in governance structures and project teams. Patient partners should be valued for all their lived experience of their condition. However, they should also be valued for all the forms of expertise they can contribute, for example, related to their studies, profession, or other forms of lived experience. And finally, of course, patient partners should be fairly compensated. There are many reasons why patient engagement matters. For example, patient engagement improves research design, efficiency, outcomes, as well as increases the value of research dollars spent. It gives voice to perspectives that would otherwise be lost and contributes to a more ethical research in research spaces. Patient engagement also leads to more equitable, collaborative culture within research spaces. Patient engagement also matters because it helps cultivate inclusion and accessibility within research. It reduces potential harms to study participants and to the ME community, and it builds long-lasting relationships and demonstrates respect for our community. Finally, patient engagement matters because it is often a funding criteria or a requirement.
So how do we actually do patient engagement? It really starts with including patients in every phase of research, from the first idea right through to sharing results. It's about making sure they're part of the team, not just invited to the table. Patient engagement can be made more meaningful by offering simple accommodations, like holding virtual meetings, being thoughtful about your scheduling, keep sessions shorter. Sending documents ahead of time helps everyone on the team come prepared without feeling rushed. Reducing prep time before meetings also shows respect for people's time and energy. And maybe most importantly, fostering a come as you are atmosphere where titles are left at the door and everyone's experience is valued equally. That's what creates real collaboration and trust in patient engagement. And establishing roles and expectations also helps to increase trust and prevent misunderstandings. Fair and creative compensation shows that the patient partner's time and expertise are valued, as well as giving credit through authorship, presentations, or recognition. That reinforces that their contributions are essential to the success and integrity of the research. So, what are some of the common pitfalls of patient engagement? Well, one is definitely tokenism, where we're just there because it checks a box and we're included in name only, without giving us meaningful roles in decision making. Another is unconscious bias, where our lived experience is not recognized as valuable expertise and is given less weight in discussions. It's also critical to provide adequate support so patient partners can participate fully, whether that's physical accommodations, accessible materials, or flexible scheduling. And finally, it must be recognized the vulnerability that's involved in when patients share their story and deeply personal or even traumatic experience to help the project. Failing to acknowledge that can be harmful. So avoiding these pitfalls is essential to creating authentic, respectful, and impactful engagement.
As the ME community knows all too well, harmful research continues to this day about graded exercise therapy, or GET, as a potential treatment for ME. And this is even though graded exercise therapy is not safe for ME patients. This research continues today and perpetuates harm by ignoring evidence from millions of ME patients globally and a growing body of scientific evidence. Patient partners can help researchers not get it wrong. If researchers are meaningfully engaged with knowledgeable and experienced patient partners, harmful research such as GET research would never happen. While we are always learning, I believe the ICANN CME Research Network is a good example of how patient partnership can be done well. Within our network, knowledgeable and experienced patient partners are integrated throughout the network governance, where they are co-founders of the network, members of the community leadership team, co-leaders, and members of working groups. Patient partners are also mandated to be a part of every research project funded by the network. They are compensated fairly and, if need be, creatively. We would like to share 2 helpful resources on patient partnership. The first is the patient-led research collaborative scorecards. This free tool, available online, helps evaluate how effective a patient group and research partner collaboration will be at conducting truly patient-led research. The scorecards focus on 4 areas to advance patient-led collaborative research projects. These are: first, patient partner governance, which evaluates the degree to which decision-making power and governance is shared between patient groups and partner groups. Then, integration into the research process, which evaluates the degree to which patients are involved in every phase of the research process and key committees, including study design, protocols, trial inclusion, analysis, and reporting. The patient burden scorecard evaluates the degree to which patient burden and associated trauma is addressed, including accommodating patients who are dealing with illness and symptoms, compensation for patients' time and skills. And finally, the research organization readiness scorecard evaluates the ability of the research organization to engage in meaningful patient partnership. This readiness assessment allows patients to discern the research organization's level of collaboration and willingness to share control. This is an example from the Patient Partner Governance Scorecard on meaningful decision-making between groups. The scorecard ranges from -2 or non-collaboration to +2 ideal collaboration. On the left-hand side at -2 or non-collaboration, regarding meaningful decision-making between groups, this could translate as decision-making for significant decisions, such as funding, study design, or publication, that are not communicated transparently, or the research organization decides the decision-making process without patient input. In the middle, it's 0 or acceptable collaboration. The decision-making process for significant decisions is well communicated and agreed upon between patients and the research organization. And then at +2 or ideal collaboration, decision-making for significant decisions is well communicated and agreed upon between patient and partner groups, with deference given to the patient group with sufficient support to make the decision.
So there are many tools and courses to help you in your journey towards patient engagement, but this course that Sabrina Poirier and I helped develop is available through the CIHR IMHA website at the link provided. This course, Patient Partnership Tips and Tools, is a how-to guide for patient engagement and research, and it is a little different because it's designed to support everyone on the research team, from patient partners, researchers, to trainees alike, in meaningful patient engagement. Accessibility was a key focus in creating the program so it can reach as many people as possible. So the course is divided into 4 modules. The first introduces the concept of patient engagement. The second covers the research process, helping patient partners understand it and helping research teams learn how to support patient partners effectively. The third focuses on setting up a research project to ensure successful partnership from the start. And the fourth addresses patient engagement in practice for patient partners. It shows how to be an active team member. And for research teams, it provides strategies for including patients throughout the project. So for more on patient engagement, CIHR IMHA offers guides that are listed here, training modules, and a YouTube video on why it matters. Like Carrie mentioned previously, the patient-led research collaborative with the Council of Medical Specialty Societies provides scorecards to help teams evaluate and strengthen meaningful patient involvement. So these are readings that highlight the importance of value and patients as full partners in research and healthcare. Of course, one of the resources is the I Can CME website, where you can find many resources for researchers. Zal Press and Don Richards explore patient ownership and the journey from engagement to equity, while Don Richards emphasizes seeing the patient as a whole person. Additionally, Don Richards and colleagues address patient partner compensation, sharing perspectives on why and how patients should be fairly recognized for their contributions. These articles listed provide critical insights from patient partners on the challenges of patient engagement. They explore tokenism, showing how patients can be included without meaningful involvement. They reflect on common ways engagement can go wrong and discuss barriers and solutions to fair patient partner compensation in research. Thank you all for attending today, and I hope this session with Carrie and I have given you a clear understanding of what patient engagement is and why it matters so very much in research.
Thank you.
Thanks, guys. That was great. Okay. So that's a great start for all of us to be able to think about patient partnership, both the patients that are in the audience and thinking about maybe wanting to get started with this, and also for the researchers that are in the audience, also a great opportunity for you to think about how could you engage more patients. And so they've given us a great list of resources and tools to get started. And we love this topic at I Can CME. So we'd be happy to help you and support you and connect you if that's helpful. Thanks so much. Okay, we're going to be bringing up Daisy again. Daisy was with us on day 1, and she was a big hit, doing a presentation from a clinical perspective of family medicine. And this time, Daisy, you're going to be addressing ableism and internalized ableism in research and clinical settings. And so welcome back, and thank you for doing this for us. And I think it's an important topic because I think if we don't look inwards and kind of unpack a little bit of maybe what we didn't have to think about or unpack, before, we definitely have to do that work now with the state of the world being what it is. And also, I think to be safe in healthcare and research environments, I think we have to unpack some of this. So I'm excited to see your presentation. Feel free to get started when you're ready.
Thank you. And thank you, Trudy and Carrie, for that presentation. It was very, very lovely and made me very, very excited and happy to see that. So I'm Daisy. I'm a family doc in Edmonton, Alberta, and I'm also a patient with some lived experience with ME, and was diagnosed back in 2020. And so as a woman with chronic illness, I experienced well, even more so, misogyny, discrimination in my workplace, in academics, personally, as a patient. And so I got involved pretty quickly in learning and unlearning more about this area, and then trying to become an advocate. So I'm not a researcher. So again, I'm glad for the presentation before as well, but I'll try my best. I do think that what I'm covering here and what I want to say is applicable to education, research, the application of clinical medicine and caring for patients, and also the human condition. So, oh, how do I, sorry, let me just, I'm looking at my Zoom screen.
It looks good to us.
It does, but I'm on Zoom and can't see. Is it advancing?
It's not, no.
Okay, hold on. I'm just going to stop sharing for a second.
Sure.
So I can get back to my present. Oh, that's why it's in a different. Okay, if we share, I apologize.
Don't be silly. That's part of the experience of having a live event.
What do you see now?
I see your slides, but I see them not shared yet. Now you're in slideshow.
Okay, perfect. Do you see advancing slides?
Yeah, I do.
Okay, Perfect. My disclosures are honoraria for ICANN CME and as a counselor for the College of Physicians and Surgeons, and I have no other disclosures. A little bit about me, I think I already covered this, but I'm also a mom and I have a clinical practice and provide home visits and virtual care, and we'll get to that later. And just some things to think about as we go on. And you might have seen some of this before. So disability, disabled, they're neutral words, and they can present differently. People may have multiple disabilities. They may be visible, invisible, different on different days, good days, bad days. It fluctuates, it's episodic, and these all make it very challenging for us, for patients, and for those who look at patients with disabilities. It increases that the assumptions, the discrimination, and it also it can be masked by the person as they want to be functional. They want to hold on to the things that they have in their life and their work and their family and want to fit in. And so it's even harder to see that way. So it's complex, and what we see is not often what is happening. So what is ableism? So a system of assigning value to somebody's body, their minds, based on a societally constructed idea of what is normal. So society, I think, would be defined as, for this, somebody, the group that's in power, the group that's making these definitions of what is normal, what is productive, what is desirable, what defines how somebody's intelligent or fit or fit to reproduce. And these ideas in our society are deeply rooted in eugenics, anti-Blackness, misogyny, colonialism, imperialism, capitalism, the ones who are in power, right? And this systemic oppression, and I think we need to start using the word oppression more in this work of anti-discrimination, anti-racism, and discussing ableism here. This leads people in society to determine someone's value based on culture, age, language, all of these characteristics that impact us all. So this comment here, you do not have to be disabled to experience ableism. That's because someone else has assigned your value based on what they see in you, or they assume they see in you. So your color can be assigned value to someone, even though you're not disabled. They may view you as lesser than or not as smart as, etc., etc. And so we all live in this together, and it depends who's in power. So this sort of reflects on that. There is no neutral body from which our bodies deviate from. So nobody stands outside the consequences of injustice and inequality. So with that, what is internalized ableism or internalized oppression? Because I'm trying to use that language. This is an involuntary reaction to oppression. It comes from the outside, but becomes something from the inside, where people in a group, they start to loathe themselves, not like themselves. They believe what others have put upon them and oppressed them with. They blame themselves. It is my fault, or, they are right. I am not good enough. Rather than believing or recognizing that it is a societal construct. And the issue with this is not only that it leads to health outcomes, but it can become externalized again when that person starts to apply those beliefs outwards on those around them. They can distance themselves from other people with disabilities, and then start to copy those same judgments and discriminatory actions, the ableist norms, and apply them to members of their own group. So external becomes internal, and then they become part of the oppression as well. So I know this is a lot going on here. I used to call this a flower of power, but here it's a wheel of power and privilege. And you might have seen this before as well. And I really like this image. It demonstrates intersectionality of multiple different areas that make up a person. And it still silos a little bit all of the different characteristics that we have here. So it doesn't quite give as much of the intersectionality overlap that I wish it would. But the thing I like about this is that it can give us an intentional, genuine guide on being curious, on analyzing who we are and who's in front of us, who's the subject of research, who's participating in research, who's asking the questions, and so on. And this is also important because it changes depending on what context you're in. So I'll use myself as an example. I'm a middle-aged, white-passing woman, cisgender woman, and with neurodiversity and chronic illness. So if we look at this with privilege and power and normal in quotes there in the middle, we see that white passing, I have more power and privilege than those who are darker in color. I have more privilege identifying as a cisgender woman than somebody who identifies more from the LGBTQ2S plus population, but I have less power than a white cis man who is wealthy. And so when I'm seeing a patient, I sit in that position of power as a physician with education, which is also on here, and with a middle-aged, white passing, and I'm sitting in front of, say, an Indigenous woman, young woman, you know, there's a power differential there, and I have to be, I have to recognize that. But if I go and then sit on my on a board, right, where they're all retired white men, say, my position of power is much, much different. And I have to take that into account again. And so does the board, right? Because the question is why? What do we do? How do we act? How does the rest of the group act to help or hinder? And how do we deal with that? So this has an impact in my care for a patient. How do I make it safe when there is an inherent power differential? How do I help address that? In research, you know, who's asking the questions? Where are they sitting in this circle? Are they in a marginalized part of this circle on the outside, or are they sitting all in the middle there? So who's asking the questions? Who are they asking the questions of or for? Who's the subject of the research? Is it the same group? Are we asking all of the questions of the ones who are in the position of power and privilege? Have we forgotten another group or groups? An example would be women being the subject of research. They have not been such in the development of lab values and medications and disease processes in the past. And we're trying to change that now. And then the other thing I like to see here is who have we not even thought of? And this can help, again, remind us that there are other characteristics of people that we might have unintentionally harmed with our questions or research or care, depending on that context we're in. And this can help us try and mitigate or avoid that or address it. And so this is something that takes time to sit with and look at. I could spend a lot more time with this, but I think I'll move on here. So internalized oppression also comes from microaggressions. These are constant, usually intangible, discriminating, prejudiced interactions, and they can become part of our internal narrative and reality, right? If somebody hears their whole life, you know, you're not good enough, I'm just going to give my personal example, it's the easiest. You're not good enough because you're a woman, right? You're not smart enough. And you hear that all the time as a child, as a teenager, when you're going to school, you're not good enough because you're a woman. You start to believe that. And you see it everywhere and you feel it in these microaggressions, and it becomes part of you, becomes part of that internalization of ableism. So why does this matter? It matters because this is reality. This is affecting patients, research, everyone, right? And it affects access, barriers to access to the research, to participating in research, to getting health care. And then it impacts the relationships. If you don't have a safe, growing relationship with someone who's caring for you, or you want to impart your experience, you're not going to be able to have open communication, genuine listening and understanding. And then we just won't get as good of a result as we would if we did. In the World Health Organization, they define health as more than just medical, right? More than just physical and mental. It's social well-being. And as we looked at that wheel of power, there's a lot of social determinants of health on there, and we need to remember them because that is health. That is our job. And so in healthcare and research, ableism and internalized ableism, oppression, as I've mentioned before, it can have health outcomes. We may develop shame, grief, that loss of what we had and no longer have because of illness, because of marginalization, etc., hopelessness, devaluation. All of that impacts health outcomes. And in research, it also shapes what questions are asked or not asked. How do we interpret that, the answers, quote unquote? How do we apply it in research and clinical medicine, policy, guidelines, and more? And if ableism is impacting all of that, health will be impacted. An example I wanted to give for my clinical colleagues here is I recently did a bit of a deep dive into diabetes research and how to give better care to patients. And the research asked very ableist questions. If you take a look, how do we improve care? Did we order enough tests? Are we doing the tests often enough? Did the patient take all of their medications? Did they do the labs every 3 months? Are they following the lifestyle advice? Why are they, or are they non-compliant, which is a terrible word to be using there and does not accurately communicate what's going on. And so, of course, if you ask a discriminatory question like that, you're going to get results that are harmful. So repeatedly, I was reading in different studies, the answer was patients are not doing labs often enough. Patients are not taking their medications. Patients are not exercising enough. They're not seeing their doctor often enough. And that puts all of the blame, all of that ableist mentality onto the patient and harms them. Nobody asked why? Like, what is the patient context? Where are they coming from? Do they even know why they need to do labs every 3 months? Why is that a goal? Why is that a question in research? Maybe we should be asking, what does the patient want when they have diabetes? Maybe they want some more hard outcomes like heart attack and stroke and quality of life. Did we ask why patients can't take their medications? Is there a financial barrier? Do they have priorities over housing and food because they have housing and food insecurity? Can they travel to go to the lab? There's so many things, and it is not taken into account in research because the wrong people are asking the wrong questions, and then the wrong advice comes out of that. And then we get guidelines saying, do labs every 3 months. And then they set these markers of quality clinical care coming from us. And it is not patient-centered. It's not patient-valued. So it leads to this, asking the right questions, looking for the right outcomes, asking what is important to the patient, and actually asking patients, what is important to you? What are your goals and goals of care? What can I know to help you better about you as a person, not as a broad stroke when I'm saying, all patients. I'm talking about asking the individual in front of you. And in research inclusion, we know that those living with disabilities are underrepresented in education, the profession, and in research. And that means those who do the research and those who are the subject of research and our patients, right? And this drives this vicious cycle of marginalization, and it's perpetuated by admissions, curriculum design and education, discrimination by professors and teachers, peers, students, everyone around them. And this all creates a barrier to getting the research done. So we need intentional, genuine, informed inclusion, as Carrie and Trudy talked about earlier, of those who are participating in and creating research, and not tokenism. And this can lead to policy change in the future as well to help reduce the barriers we just talked about and maybe break that cycle and start a better cycle. So what can I do? What can we do as clinicians too, and as humans? Practice empathy and self-reflection. Checking your biases and checking on your gut feelings. And before you step into the room to see a patient, how do I feel? You know, do I have any biases that I need to sit with and then acknowledge, right? If we don't acknowledge them, they can take over and inform our practice negatively, right? We have to acknowledge that grief that might be present over a loss of function, quality of life. We also need to remember that disability does not mean less than or less intelligent. It doesn't mean a psychiatric illness. It's not a psychiatric diagnosis. We've talked about this before. Just because we don't know what the diagnosis is, or the profession or science doesn't have a test, it does not mean it's psychiatric. It puts it right back on the patient because we can't take ownership over helping and learning more. So if we don't know, we need to check why and take that action, right? Everyone has bias. We all have it as a human being, and as physicians especially. It does not make us a bad physician to be uncertain or not know or even make mistakes, because we're human. But what defines us is, what do we do next? What's our action, right? And our actions are in part defined by our biases. We need to address social media as well, because we have turned away patients by dismissing them, not validating them, not listening, as I said in the last slide as well, misdiagnosing them as having a psychiatric disorder. I was told the same when I presented with ME. There's a reason our patients are looking for validation and information to advocate for themselves in a system that's oppressed them even further, right? And social media can be good and it can be harmful. There are people on there, organizations on there just trying to make a profit and not actually help our patients, but there are also a lot of very good, credible resources on there, and we can help them navigate. We can even learn on social media or even take part in it and post stuff on social media to help patients too. So it's not black and white. Social media is not a negative, and we need to feel or analyze how we feel when a patient says, you know, I saw on TikTok, I have this. And we need to check that reaction and then realize maybe why have they gone there? It's because we need to do better helping. So providing more accessible clinical care, this is under our control as well. And this is in research as well, as Carrie and Trudy mentioned, with accessibility, shorter appointments, virtual appointments, recording. I know a lot of clinicians and physicians are worried about the recording aspect. It's legal if only one of the two in the party are aware of the recording. So it can happen even without our knowledge. But let's put it out there and say, maybe you need to record this because this is a lot. And, you know, right now, maybe you're especially having difficulties paying attention or having more brain fog, or this, you know, you're in a crash. Record so that you can look at it later, listen to it later, help with your family or caregiver so that they can also listen and learn and have questions for next time. change your environment as well so that it might be lower light, or I like to turn off all the lights in my exam room and just leave my computer on so that serves as kind of an ambient light, or leave the door open a crack and let my patient lay on the bed with their eye coverings or sunglasses, and that's how we do the visit. That's okay. Different doesn't mean worse. And we can still practice medicine in a slightly dimmer environment. And if we need to turn on the lights briefly for a visual examination, so be it. I do think cancellation policies are punitive for the patient and increase barriers. I don't do them. Instead, again, let's have that kind curiosity. Why is our patient always missing their appointment? Maybe it's because they don't have transportation, or their paycheck didn't come in and they can't afford transit today, or they're relying on a source of unreliable transit because they live in a place that does not have that. We need to ask why. In the end, flexibility, and this applies in research too. Ask others, not just the patients, but anybody who's informing your care or your work or your guidelines. What do you need so that we can do the best work we can? What can we do? Be flexible. Again, different is not worse. And this leads to advocacy. So advocacy, I'm told, it's not in your lane. It's political. That's not your job. Stay out of it. Actually, advocacy is a formal competency in our education. It's in our CanMEDS criteria. Here's another flower for you. Health advocates in there. We know there are social determinants of health. It's our job. It is our lane. It's built into our education. We need to do it. And advocacy can take place with a single patient in front of us when we're doing paperwork, disability insurance forms, or on the political stage when we're asking for recognition of non-psychiatric complex care remuneration. This is important and it's part of our job, right? And advocacy can be research. Research can be advocacy. Research can help define or prove or bring to light a problem most of us probably already know exists, but it needs to be out there, and that can shape guidelines and policy and say, you need to make changes because we have shown you there's a problem. So we need to listen genuinely, intentionally, ongoing throughout the process in education, research, advocacy, everything we do, not just in our office with our patient. And overall, not just in addressing ME, but all patients, and maybe again, the human condition in general when we approach it, let's approach it with some humility, compassion, curiosity, kind curiosity, reflection, self-reflection, and kindness. We are all continually learning and unlearning. We need to learn about our biases, our internalized ableism, how that affects our practice and our education and our research. And it's okay to say, I don't know, but we need to follow it up. Some resources, disability paperwork resources, Bateman Horn Center, and so on, just in general. And then I have some questionnaires, because again, we talked about how disabilities can fluctuate or be episodic, and then we need to know about it, not just see what's right in front of us for that brief, hopefully not so brief anymore after this talk, but assessment of the patient. And I've done a couple interviews on medical gaslighting and how it's impacting patients, how stigma impacts patient outcomes. And there's actually a follow-up on the paper on the right here. Dr. DeMatt published another paper, I think last month, saying that the long COVID patients who had higher rates of stigma in his initial study have worse outcomes now. So why is that? And then some suggested reading and viewing in general. And again, this will be with the slides later. You can take a look at that. Okay, I think that is all.
That was so wonderful. Thank you so much for doing this. That was a great presentation. I long for the day where this is sort of common knowledge. I can't think of a single person that can't learn something from what you just presented. I'm constantly trying to think about these things, and I don't hold a position of particular power, and I think it's still important. So I would love to see doctors and researchers think about, how is it playing in their environment and what can they do to lead the way, to model, to be good examples. So I think it's, yeah, such a great topic, and you did such a great job. Thank you so much. And you'll be back for our panel? Excellent. Okay, we'll ask some questions then. And our last presentation of the entire conference, because we'll have the panel after that, is Tess Fowler and Isabel Ramirez Burnett, who are, sorry, switching on the screen in front of me. It took my brain a second. Whew, I'm tired. But I am so thrilled to see you both. Thank you both so much for doing this. This part of the agenda had a bit of a theme of patient-driven, patient partnership. And so that's why we placed you two in here, because some of the work that you're doing, I think, really kind of brings to life some of the things we're talking about. So I'd love to hear you guys talk a little bit about your lactate research, patient-driven lactate research, and then we'll move on to the panel after that. And again, their bios are going to be in the chat and in your conference program. Welcome.
Right, thank you. All right, screen is coming up. Thank you very much for having us. Thank you for the previous presentation, Dr. Fong. That's a great segue into what we're doing here. And thank you for so many of the things that you said that are pertinent to patients and patient-led research. And today, the two of us, Tess Fowler and myself, Isabel, we are with Renegade Research. We are a patient-led nonprofit 501c3 that focuses on agile research and also education and care for patients with ME/CFS, long COVID, and associated comorbidities. And although the two of us are presenting this today, we're just the voices for a larger team of patient-led and researchers, some of which are also patients that did this work. Next slide, please. Okay, so for our agenda, we'll go a little bit into lactate, the science, how the acid test, which is the label that the patients gave to this project, started, the objectives of the study, how it was conducted, some of the results and conclusions, and what are the next steps. And next. So the patients and researchers and patient researchers in here are Sierra Wright from Positive Nutrition, Naomi Harvey, who is a volunteer at Renegade Research, Harriet Carroll with Lund University, Todd Davenport at University of the Pacific, Tess and myself, and David Petrino at Mount Sinai. And of course, the participants, 131 people across 20 countries who we lovingly labeled as the acid testers. Next. So in here you see these two graphics. On the right, we're looking at normal levels of lactate at rest, which is generally under 2 millimoles. And this is what you expect to see under normal circumstances, under normal oxygen utilization at rest. But on the left, it shows that when oxygen utilization is impaired or mitochondrial function is limited, pyruvate is then diverted from aerobic energy to lactate production and switch into anaerobic metabolism. Next. And then we also want to acknowledge some of the previous work that was done in lactate specific to ME during 2-day CPET, comparing ME/CFS patients to normal but deconditioned controls. And we can see in some of the research here where ME patients reach that lactate threshold sooner then the controls, and then on the next day, the MSEF, the healthy controls dispose of that lactate more efficiently. Next. And some of the fun aspect of this is also how it got started. So again, patients who are always discussing symptoms, observations, the science of everything that has to do with these conditions, whether that is on X or other platforms and forums. Patients always reading the research and commenting and bringing in their expertise. So in here we can see one of the early posts from Ciara, where she says, looking at lactate, new microbiome tests shows high lactate producers. So Lactobacillus eficalis and high lactate users, Akkermansia, and so forth. And also discussing again some of the science that was going on and looking at hypotheses of inflammatory acid-base disruption that may be underpinning long COVID. Next. And in the development of the project, so creating a protocol, how would the patients be testing, what lactate meters, getting funding, getting those devices and shipping them to patients across the world, literally. Patients doing their self-testing and then sharing the results once again on X and also on formal surveys. And if you remember from a couple of the previous slides, the normal levels are about 2, up to 2. And in here we can see elevated lactate measurements of 5.8, 4.1, 14 in one. One of mine was 9 something. So we can see that, that hypothesis tracking. Next. You're muted.
Hi, I'm Tess. So I've been an ME patient for 20 years now, but also have a research background. So yeah, the acid test, it was really exciting times on Twitter. It was really fun how it started from patients just sort of discussing these ideas and then having it turn into an actual study really quickly, like within a month or 2. So the objectives of the study were to demonstrate that we could do this decentralized research, just to demonstrate the feasibility of it with the decentralized research, which you've heard a lot about in this session today. We were hoping to recruit more functionally disabled patients and wanted to measure lactate after a bad day, look at correlations with demographics. And I'll mention for recruiting more disabled patients, a fun anecdote was somebody on Twitter who was severe actually went and created a video of how to do this lactate testing. And so there were just a lot of patients contributing however they were able to. It was a community project, which was really amazing. Okay, so what we did was we used these at-home lactate monitoring devices that Isabel showed the photos of. You just do a finger prick and get a nearly instant reading of the lactate levels. People did it right after waking, when they were fasted, and then an hour after a light breakfast. Todd Davenport was really great to put together the IRB for us. And he hosted a survey where people were able to go take the data they were collecting themselves at home and submit it to the survey so we could collect all of it. Okay, so we had 131 respondents from all over the world, 20 different countries. This is a bit of the demographics of the participants. I'll just kind of point out that the majority had a disease duration of between 1 and 5 years. So, likely that long COVID cohort. Also on the left, you can see what caused the disease onset. And again, you can see about 50% were from COVID. The second there was herpes virus infection. looking at the severity levels, is mostly moderate to severe. You can see about 50% of the people had moderate to severe symptoms at rest, severe symptoms with exercise, and then a smaller number were mild, and some on the severe end. So like we mentioned, this was all coming together on Twitter. So patients, as they were doing this study, were going and posting their results and having discussions of it, talking about the science, talking about what they were finding. And I'll just point out here in the middle one, the words enlightening and validating. So for a lot of patients, they had never had abnormal test results before, and they were able to do this at home and show, look, I'm getting 9.1 millimoles per liter. But it wasn't everybody, not everybody had the elevated lactate. So you can see the one on the right. So someone said, yay, seems to be perfectly fine, yay, but also a bit disappointed. So just kind of pointing out from the patient's perspective, this was just really interesting, really fun for people to do. All right, so the actual lactate measurements, one of the keys here is that looking at the lactate on waking, the resting lactate following a good day, following versus a bad day, the bad day was significantly higher. When we're considering these results, so there's some confounders and limitations. Todd Davenport was just pointing out that when you're severe, you might not actually have a good day. So when patients are inputting these results, they might not ever tag that this was during a good day. So you're kind of not really capturing, the severe end. There's also noise from home-based measurements. Like not everybody used the exact same devices. The majority used these GlucoRx devices, but there were some other ones like the Edge. And then there was limited control over factors like diet. So the survey did collect information about different medications and supplements, things that people might be doing that would modify their lactate. But again, it's kind of hard to control for that. So Todd and Kira actually gave a really detailed research roundtable for us. And it's posted on our YouTube channel where they really go into a lot of more of the specific results. So if you're really interested in more of the results of lactate measurements, you can go there. But just to point out some of the highlights here, again, the resting lactate was higher after a bad day. This decentralized citizen science approach is feasible. It worked. We were able to do it completely decentralized, doing these at-home measurements by patients. And abnormal results were validating for the patients. So some of their implications, the results reinforce mitochondrial anaerobic dysfunction in ME. We can run follow-up citizen science studies using this design. And also there's a potential for interventions. Related to the interventions, again, on Twitter, patients started to, on their own, sort of experiment with interventions and seeing how it affected their lactate. So this wasn't part of the official survey. We weren't collecting this data, but I just thought this was interesting that patients sort of running their own self-experiments then to follow up. And this was one where you can read here, but guys, this is working for me, trying some supplements. I'll pass it back off to Isabel.
Yes, and for next step, so a paper is in the works, and also looking at running other studies with continuous lactate monitors that are now hitting the market, and also how lactate levels respond to interventions like things that target PEM, ALA, thiamine. And in my own experience, I can see how tramadol, which is prescribed for pain, predictably lowers lactate levels for myself and others who have shared their anecdotes as well. Next. And again, Renegade Research is a patient-led decentralized 501c3. We're running other decentralized projects. We invite everyone to reach out and inquire, collaborate. This is a list of some of the projects that we have coming up at different stages of development. Something like photobiomodulation with via light devices, a formula for PEM, another phase of the nicotine test, and more. Next. And as Tess mentioned, there is a presentation on our YouTube channel, youtube.com/Renegade Research by Kira and Todd, with more detail on the project and more detail on the science of it. And to follow up on all of the other patient-led projects that we do, you can sign up for our newsletter on our website, renegade-research.org, and follow us on X and look more at these anecdotes following the hashtag #TheAssetTest. And thank you for having us today.
Thank you guys so much. That was a fantastic presentation. I'm going to invite everybody back to join us for the panel. Just turn on your computers and join us, and we'll get everybody set up here. Welcome back to everyone. My name is Sabrina, and I'll be moderating this panel in case you didn't see enough of me yet. We're almost done, guys. We're almost done. We're at the home stretch. Please read our bios in the conference guide so that you know who we are. The title of today's panel is Collective Deep Thinking to Move ME Research Design Forward. So we don't want to stay stuck anymore. We don't want to stay where we are. We want to talk about how we move everything forward, because we've been stuck for a while for a whole host of very important and good reasons. But if not us, then who, right? It has to be like we have to come together under the same roof and have conversations that are tough and figure out how we move things forward. And that's kind of what we were trying to accomplish with the beginning of this conference, I think. And so this panel is kind of going to be asking the question in a moment, like, where do we go from here? We feel it's really important because there's some significant gaps that we keep seeing repeated in research. And we may be seeing more and more confusion and conflation in the data, exclusions of entire groups of patients in research projects, and at times possibly poor results and increased dropout rate. Whether we're tracking that or not, we're definitely seeing it. So this is an opportunity for us to see what we can do about that. So thank you all for being here today to present, and thank you for being on the panel. And I'm just noting the time so that I remember what time we started, so I can remember what time we should finish, because I'm very tired, as I'm sure everybody is. I'm going to start off with just a really simple question to Tess and Isabel, which is, when you guys were doing this research and it was patient-driven, and I know that you've kind of collaborated with other researchers and you've been in other spaces, what's the difference between the two sort of environments or approaches, and which one do you like better?
You want to go first?
You're
on
on
mute.
mute.
It's not letting me unmute. There we go. I was going to say, we've actually been really fortunate to work with amazing researchers. So I think that we've had good experiences more working in the academic setting, but definitely these decentralized projects, patient-led, are a lot of fun. You want to add to that, Isabel?
Yeah, I would agree. They are a lot of fun, but it's also really the difference is that, with the patients bringing their experience and even ourselves being patients, there are things that we don't experience that other people do, right? That perspective is brought in. And as Tess said, we've been really, really fortunate to work with academic researchers and collaborate with people who are open to that feedback. And for the most part, the people that we've experienced working with really have the drive to include the patient perspective and all of the different considerations that need to go in there. Sometimes, or a lot of the times, it is confounded by funding to make a lot of things happen that need to happen to move all of the different parts, right? But I think that from the academic perspective, if they keep that open mind, and we were talking today about, being scrappy and looking for ways to make it happen. You may need to, decrease the number of participants, or you may need to collaborate with another center, whatever needs to be done, to maximize that inclusion to the extent possible. Then look for those ways, because it's also fun to uncover all of the different things that can be done.
Yeah, I think that the inclusion that's really important. Like we were mentioning during the talk, that as patients trying to make this study or other studies happen, we're very aware of how difficult it can be for patients, and so we kind of go out of our way to support people and make things as clear as possible. And I think that was like I mentioned, that was the whole community doing that for the acid test. It was people coming together on Twitter and explaining things to each other, helping each other, find the protocol and learn how to use the glucose devices and everything.
Yeah, our community is pretty amazing. Like, I mean, shout out to the community, because we don't do that enough, frankly. We have a really brilliant community. We have a really hardworking community. It's a community that I'm always angry when I hear people sort of comment about, oh, it must be so nice to stay home, and it must be so nice to be, resting. And it's like, I haven't met a single ME patient who's not wanting to do what they're meant to do, what their calling was, whatever that was. And on top of that, they go out of their way to help each other. They go out of their way to try and learn about this illness. And we don't spend enough time, I think, giving kudos for that, and also for patient partnership and advocacy. I really think that our community does a pretty phenomenal job, given the huge mountain it has to climb to be able to do any of this work. And I see so many faces right now on the screen that are doing like this tremendous heavy lifting. And I just want to say kudos to those here, but also all those who aren't here and who are doing other things right now and contributing to the community in other ways and couldn't be here because they were doing that work, or they were doing it last week and they're crashed. I think we have such a special community, and I wish we could spend more time talking about that and lifting that up, but we're also busy working. So, I saw a real shift when you guys were doing some of the projects around the nicotine project and the and this one, where all of a sudden everybody felt very empowered because they had the tools to kind of measure things themselves. And it was fun to watch the tweets going by. And so some of the ones you showed actually are the ones that I was like, oh, this is like really nice. So actually, I think patient partnership is what we're calling it in Canada, but it's that partnership of like clinicians, researchers, and patients coming together and being able to do the work. We're calling it patient driven with you guys, but it's all important. And I think however we come to that, everybody has a role to play. Everybody's important. But for too long, patients have been kind of to the side of that process. And I think the more we bring patients in and the more that we see patients driving it forward, what a difference. Like, I'm just seeing huge shifts. And then great researchers like Alain and Todd and others who can't be here right now are also a big part of that, because they create space. And, going back to Daisy's presentation about power dynamics, it's all good and well if patients want to get involved, but if researchers and clinicians can't create the space for them to be included or can't make that experience accessible, then we're not going to get very far. Daisy, going back to your presentation, how are doctors learning about ableism right now? If I just said to you, are they getting that training anywhere?
So I did not. I graduated from med school in 2010, just for context, but I do know that the current generation of medical students are learning about it. They're learning about Indigenous health and social determinants of health in a lot more detail, and even speak the language that my colleagues do not in the anti-discrimination, anti-racism space. So I have hope. It's just, as we talked about on the other days of this conference as well, it's so slow. It takes so much time, and we're looking at like a generation away of fully fledged physicians who won't automatically put up their defenses when they hear the word ableism, or they hear the word or the phrase, examine your biases, right? But it's getting better. My focus is mostly on the staff physicians and clinicians who will not get this education or this exposure because they won't seek it.
Yeah, and we're never going to reach everybody. I always say that to people. We're never going to reach everybody, but there is a group of people out there who, once they know that there's potential to discuss these things or to learn, they're open. And that's the ones we have to aim for and eventually reach tipping point, I hope. Jacqueline, can I ask you a little bit of an off-topic question following on from what I just asked Daisy? In your profession, are you guys talking about anything around these types of topic around ableism and like what chronic illness looks like, what invisible disability looks like? Like, how are you guys dealing with it in your field?
Absolutely. I mean, we have a lot of training because my field is rooted in psychology, right? And so a lot of what we learn even in graduate school is about, combating these types of things. That said, it doesn't mean that it doesn't exist in my field. And to the contrary, I think that there's a lot of skepticism because people view things from a specific lens, right? Which is that of psychopathology. And so sometimes it's hard for people to depart from that, I think, when there's other kinds of evidence. I think one of the disconnects is that, our fields tend to work in silos. And so when you come from a psychology background, you may not have worked very much with people with chronic illnesses, right? And so there is a lot of, like thinking about the biopsychosocial model, there's a lot of training in the psychological model and the social model, and not a whole lot on the biological aspects. No psychologist will tell you that, but that is just the fact, I think, from our doctoral training. I will say, though, that myself and a few of my colleagues have been very active in sort of the education space. So I've worked closely with the American Psychological Association and the American Psychiatric Association and have given several webinars and talks on post-viral syndromes. I also teach actually at the medical school to medical residents at Mount Sinai. And we teach post-viral syndromes in general and sort of how to identify them and manage them in an evidence-based way. And even just teaching them sort of about how to respond when labs come back normal and sort of, where you go from there, and that, these are diagnoses of inclusion, not exclusion, right? So it's not like, oh, well, this is, we don't know what this is, so it must be anxiety, right? I think that a lot of physicians are actually very well intentioned, and especially like the younger ones, I think, will come into care for a patient and have this model that they were taught in medical school to fix and to treat and to diagnose. And if they're not familiar with the conditions, and they have to name something, well, I don't know. They don't like to say, I don't know what this is. So it must be, anxiety or whatever. And obviously that's not helpful, and that's actually harmful for patients. And so there's a lot of education now in this space. I've also been working with the CDC and CMSS. There's a website that I can send you, Sabrina, later, but we're doing sort of national physician training on like long COVID and other infection-associated chronic conditions. And so the hope is that we can develop like a repository and resource toolkit that physicians who don't know about these conditions can very quickly read about and learn about them. So there is a lot happening in this space, but like everything else, it takes time. So yeah.
Awesome. I mean, even in our patient community, I think there's still a lot of learning, right? Like it's that internalized ableism that society teaches you, and you don't always know it's there until you get sick. And then it's that like, I should be doing better. I should be trying harder. I should be more positive. I shouldn't need that wheelchair. I should get out and walk today. You know what? I just need to push myself. It's those kinds of things that we've been trained to do. And when you're healthy, you kind of get away with it. And like, there's not a lot of payback. But when you're a sick person, that kind of thinking is so damaging. And you don't notice it until you're sick yourself and you have to kind of unpack your own internalized ableism or your internalized misogyny or your internalized whatever you've been taught that's been unhelpful. And so even in our patient community, it's hard sometimes to see new patients come in and they're so hard on themselves and sometimes hard on each other. And we have to kind of step in very gently and remind everybody that, you know, that toxic positivity is kind of what society tells us we have to be, so that we don't have to actually deal with the systemic problems that have put us in places that we are, without the supports that we need, without the access to care that we need, without the access to food or housing, or like, enough blaming the patients. And I'm not blaming the patients. I'm saying this is a societal problem and we're all suffering with it. But our community needs a lot more support and a lot more education and a lot more love. And in the absence of that, you know, this is the stuff that kind of we're dealing with. So we also have work to do, but you know, until we can provide more supports and more access, it's really kind of not the top priority, I guess, sometimes for us to be unpacking our own things because we're trying to get to the next doctor's appointment where we're going to be gaslit and told it's not that bad and it's not that serious and we should just get over it. So, you know, we're dealing with that. We're constantly in a survival sort of mode, and we have a lot of work to do, which is why, you know, in Daisy's presentation, when she's talking about that power dynamic, I think it's so important that we talk about that we acknowledge that. I think it needs to come from people like Daisy, because people like me, no matter how we may present in this environment, when I go into my doctor's office, I'm like a little church mouse. I'm so sweet. And I don't push back too much because there's too much power in that person's hands. And so I have to very carefully navigate how I say something. Whereas, you know, peer to peer, you guys can say things we can't. So for everybody listening, and I know there are some doctors listening, and I know there's some researchers listening, because I looked through the list of names multiple times today and I saw who you were, and I'm remembering who's who. But I'm going to ask you to really use your voice more. Like, go into those environments and talk to your peers and say, we can do better. This is not acceptable. And when they push back and they say things that are ableist or sexist or misogynistic or whatever the case may be, because we are in that kind of society and that can happen, push back. You know, we have to start calling this stuff out. If we just kind of let it be and we're all polite all the time, guess what keeps happening? Harm. So I've started gently and respectfully and with love calling out more of the people around me when I see this kind of stuff. But again, I have no real power in this stage of my life, so I'm asking you guys to use yours. That was my little rant for today. I promise to be the only one. Carrie and Trudy, I want to come to you guys. So you guys talked about patient partnership, and I have watched you both in the patient partnership space and have the pleasure of working and collaborating with you both in this space. And while there's lots of great stories we could tell, and you guys, you know, use the example of ICAN-CME, which we're still learning. We don't have it all figured out, but we're doing some good stuff. I would like to ask if there's anything you'd be comfortable sharing that maybe would be a little bit, you know, more realistic to kind of what the typical experience is, because I've done this work a lot and I could count on one hand probably how many times I've felt truly included, truly supported. So I'm wondering if there's anything, you know, without naming names or giving anything too much away, was there a time where you didn't feel that way, and how did it leave you feeling about the idea of being in these spaces?
For sure. Do you mind if I start, Trudy?
Oh, you go right ahead.
You were talking earlier about ableism in like clinical settings, and I think it also happens in research settings. And sometimes I mean, most of the time it comes from really good intentions, but I've often been told, "Oh, you're so sick. Like you possibly couldn't be doing this to support the research project. Let us decide, and you know, you're over here and we'll, you know, you're sick." So we just, and sometimes I just get so frustrated about that form of ableism, which is really, can we decide what we can do and what we can't do? And maybe ask us instead, what supports do you need to do this? You want to contribute to the research design and the discussions and the meetings, but like the meetings are at 7 o'clock in the morning. Let's ask me when we can hold the meeting so that I can be there, and it's not going to take away from hopefully making supper for my child that night, you know? And so, and I want to be the one to decide what I'm willing to give and not willing to give and have that decided for me. So I think that's kind of maybe a pet peeve for me, for sure. And I think sometimes being the only patient partner, it's a simple one, but it's hard to be the only patient partner in the room. It's impossible for me, especially as like a white cis woman with moderate ME and a high education, to represent the spectrum of the illness and of the experience of being sick. And so, I mean, what I try to do often is, if I get a foot in the door, I try to bring other people in behind me. So I'll take the seat, thank you, but I'm going to work really, really hard to make sure you understand that I don't want to be the only one there. And in fact, maybe there's even better people who should be there than me. And who can I bring in who can bring more experience to this project? And maybe that links to perhaps the lack of representation in general, the diversity of people who are, and it comes down to privilege in many ways. I understand it. I'm lucky enough to have insurance. I'm lucky enough to be moderate. I'm lucky enough to have a background in research so that, it's easier for me to be there because I'm able to feed my child because I have insurance and I'm not using my energy to try to work part-time to make sure we have a roof over our head. But recognizing privilege, but also making sure we make spaces for other people with different experiences of privilege, I think is really important for me.
That's an excellent point. And I would agree with you. I think privilege is playing a lot into it because there's a lot of wonderful people that we work with in small bits that we want to pull in and work with more. They're struggling with some very significant, very real things right now that they need to have more support than they have. And so we are patiently waiting for the right moment where we can pull them in, acknowledging and understanding that what they're up against is like significant and present and urgent right now. And so, these are these systemic machines that are at play that we have to be patient with the people that we're trying to engage.
100%. And I think compensating people, patient partners fairly can be a form of a bridge to help people of different experiences to be there. But there's also systemic barriers to compensation. So it's complicated. I don't know, Trudy, did you have anything else you wanted to add?
Yeah, sure. I have, good question. I have experienced a lot of tokenism over the years. So I've been involved with patient engagement for 14 years. And especially originally, it was much, much worse than it is now. I think that the CIHR making it a requirement to educate yourself on patient engagement and sometimes the requirement of a grant has actually opened the door more for people like us. But yeah, tokenism's a big one, especially when they don't even bother to hide it and say, I have to have you here because the funder is insisting. And I'm like, yeah, I don't think I'll take this project, right? And like Carrie, I guess I've got the experience now that I'm like, I have something else I have to do. So I was telling Carrie last night that I looked at projects I was involved in. And I thought, if I haven't made a difference in the past 2 years, I'm out of here. It's because I've got lots of things to do. So, and the other thing is kind of the opposite to what Carrie was saying. I was on something to do with EDIA. And I was asked as a white privileged person why I was there, why I thought that I actually fit there. And it wasn't said very nicely. And I said that I represent. So I said, you're a good example because I represent an invisible illness. And that's why I'm here, that you don't need a wheelchair or something else to be disabled. So I think that's a mistake a lot of people make. It's if you can't see it, you don't believe it. So people with invisible disabilities, the ME community included, lots of people face that discrimination. So, and I wanted to follow up on something you said, Sabrina, talking about encouraging racism. And I have friends who are Indigenous and African Nova Scotian, and I've had conversations with them, and they say if you experience racism and you don't say anything, you just walk away, or you see it posted on Facebook and you don't comment, then you are a racist yourself. That no action is the same as racism. And I learned a lot from them. And so it has made me step up when I experience sexism, racism, any ism. I try to step up and say, you know what, that's not cool, and that's so wrong, or whatever. Whatever's appropriate at the time. So I just want to follow up to what you said,
good.
because it's important for people to know that staying silent is just as bad.
Oh, there's nothing worse than being neutral. You're
Mhm.
siding with the oppressor every time. So we all need to
Yes.
make sure that we're understanding that there is no such thing as neutral. You're choosing a side always, and you have to make sure that you're choosing the right side.
Yeah.
Which brings me to another point, Trudy. You and I have talked a few times about one of the challenges we found in patient partnership has been sometimes it can be difficult because you're in a structure of power and you don't have it, and you're coming into somebody else's space, usually a researcher or a clinician space. And sometimes if there are multiple patient partners, one of the things that we can sometimes see happen is a deferring to authority
Yeah.
that you and I have talked about so many times, that it's really hard sometimes, because if you're sort of the one that's like, well, no, this isn't okay, and everybody else is like, oh my goodness, this person is just so, perfect in everything they say, and it's like it can be hard sometimes to like work collaboratively with the researcher, the clinician, and the other patients to be able to make change if everybody's deferring to the authority in the room and we're being weaponized against each other, which a lot of researchers that, we're currently working with, by the way, the ones included, are not weaponizing us against each other. But unfortunately, I have been in rooms where I've had to walk out of those rooms because we've been weaponized against each other. And I refuse to be used that way.
Yeah.
And one of the things that got attention at the beginning of the conference was the fact that we explained that we explained in the beginning in our opening remarks that we didn't want anybody to misunderstand how sick we were, because we would look okay on screen, but you wouldn't see everything that was going on. And we really felt it was important to explain how many accommodations and adaptations and all the things that we had to do to sort of be able to show up, because we didn't want it to be weaponized against our fellow patient. We didn't want anybody coming to this conference and saying, well, if they can do that, how come you can't do this? Absolutely not. I refuse to be used that way. The team refused to be used that way. But I do think that's a common problem that we see in the space with patient partnership, where some that are newer to it are sometimes drawn in by the, finally being validated and accepted and kind of, and then they kind of forget that we're all there to represent and we have to work together, and loyalty is always to the patient community. Like much love to my researchers and my clinicians on here, but loyalty is always to the patient community. Those are the people who will pick you up when you're down. They will dust you off when you need dusting off. They will help you. They will send you a lactate monitor. They got your back. So, I mean, there's never a time where we can forget that. And I think that's an important thing in patient partnership too, is that as new people come into the space, let's collaborate and work together, and we can all support each other and get along together and create that safe environment. But that deferring to authority is something that's sort of that internalized ableism, internalized patriarchy, all of it, right? And if we don't unpack it, we recreate it. So
And it comes from a good spot, right? Like people are, I mean, I'm new to chronic illness. I'm from the long COVID wave of ME that happened. And when you first get sick, you lose everything, right? You lose your identity, you lose your career, you lose your friends, you lose your partner often. And then someone invites you into a space and wants to listen to you and hear your experience. It is validating. And it's so hard, I think, and I think that's why it's important to have like knowledgeable and experienced patient partners depending on the project, because with time we learn that we're there to represent the community, right? Like Sabrina says, and it's really hard. I mean, sometimes I'm in a room and I have to stand up and say something that's really tough to hear from other people, for the people in the room, and I'm shaking in the background going, oh my God, like it's scary. My heart rate's 158, and it's terrifying, but you do it because without it, what's going to happen to the project? It's going to cause potential harm. So, and I think like, just to finish up with my first comment, but like one thing I was thinking about was like a lot of really small things sometimes go wrong in projects. Like one of my pet peeves is, oh my gosh, we so want you to be a patient partner. And I said, great. Can I where are you at in your process of creating the project? And they're like, so here's our protocol. We've gone through ethics. And I'm like, right. So I'm going to have some comments on this protocol and what happens if it has to go back through ethics. And there's this like long silence, and generally it doesn't go back through ethics, and it's already been pre-decided. And that's a little pet peeve of mine, because if you're going to ask us in, I think that there's a lot that we can say about study design. We learned about that today. Patients are brilliant and we have a lot to say, and we have lots of thoughts on study design to make it safe for our community. So
Yeah, that's a really good point. So bring us in earlier. And if
Yes.
if you missed the window, if you didn't know about patient partnership at the start of your project, that's totally okay. Bring us in whenever you can. But then let's start talking ideas for the next project and
Yeah.
let us be a part of co-creating that. And when we do give you feedback, go back through ethics. It's not that big of a deal. It doesn't take that long. And really great researchers are doing that with us. So we know it's possible. So when you tell us you can't, we know you're not telling the truth. So I want to come to Isabel, and then I'm going to shift focus a little bit. Isabel?
Yeah, just quickly, I just wanted to say, for the people that do hold the power, shifting the culture of whatever organization you're in, like clinicians can reserve appointments for patients who can't wake up early and need the times in the afternoon. And the same thing with a research appointment, if it's not able to be done in a decentralized manner, having, appointment different blocks reserved, depending on where people's ability to be able to have the better part of their day, or holding a space as a backup, getting an extra week of a backup for people who couldn't make it on their original appointment because they were ill. Just things like that don't really have a lot of extra cost, but you can set that tone and facilitate that.
That's such a good point. Yes, absolutely. And I think when we send out Daisy's presentations, I think they're going to go a long way towards some of this. She's got a lot of really practical, easy, low-hanging fruit kind of things in that presentation that you would just think would be like kind of like, oh, well, everybody's doing this, but nobody's doing this. And they're really simple, easy things that often don't cost any money. So if somebody wants to make change, if you want to have impact, and by the way, there's going to be a survey that's going to pop up when you close the screen today, please complete it. And one of the questions is about impact. We don't want to do a conference just so we can all talk to each other. Like, no offense, but I mean, I can talk to any of these brilliant people, right, without putting on a conference. We want a conference because we want to create impact. We want to spark a bunch of lights, and we want to see what burns, and we want to like light the whole world on fire with like joy and potential and, imagination of what we could be doing here. So please think about where you can have impact and where you can use this information. I do want to shift a little bit. So we've spent the first day kind of learning about level setting about where we know ME to be now, and a lot of the things we needed to kind of understand about ME. Day 2 was focused on comorbidities and less studied pathologies, and kind of some of those areas that we could be doing a better job of tracking, measuring, understanding, caring for. And then in today's discussions, we were really looking at research design and saying, okay, so we know all of this, but now what? And I think we're at the now what part. So, I would like us to think about what are some things that we could do. And I want us to keep the answers as practical as possible, because there are some students in our midst who are at the beginning of a career and they want to do it right, but they need that, like, what's that one or two tool I could add in? How can I make this protocol more inclusive? How do I make sure I can bring them into this research environment? So what are a couple of things that we could give them that would be really practical and really easy? And then we'll talk about some of the things that are actually going to take us money and time and a lot more consensus building, because I think those things are on the menu as well, and we have to talk about them as well. So let's start with some low-hanging fruit, simple things that we could do to make research design immediately better. Whether you're a patient partner or you're a researcher or you're a clinician who's working in that environment, what could we be doing? And Todd, I'm going to come to you because I've ranted at you so many times about this stuff that I feel like I know what you're going to say.
I was kind of hoping you'd ask somebody else first, but I'll take a stab at it. Thanks for the chance to address the question. There's a lot of promise in a lot of the tools that are emerging. Many things that are new, permitting new types of research designs and so forth. And as you mentioned, some of those are things that are relatively kind of still aspirational. We're still trying to kind of get our hands around what they mean and how best to use them and in whom to use them. I think probably the first thing we can do is consider where we came from as a platform to where we're going. I don't think we throw out everything we knew before, even though it's not optimal, and we're all trying to move in different directions, my group being among them. But that we take existing technologies and existing resources and we validate the new resources against them. So we really understand them. And in particular, we really understand the influence of exertion. Because really exertion is central to all of this. It's the central to the lived experience, as I've mentioned. It's central to the diagnosis of ME in a way that's very specific and really identifies a very specific patient cohort. And so unless we really sort of think through the role of exertion, how new technologies, new strategies line up with existing forms of exertional testing, then what we'll have is new stuff that has promise. And so, and all of that is out of the box. We have stable technologies and we have stable foundational material we can go from, and that way we can be sure that when we leap into some of these new ways of doing things, that we can be confident in the direction that we're jumping, and as we all go along with that.
Can I add one more thing that I think you might agree with me on?
Sure.
One of the mistakes that I'm seeing is that when people are using the DSQ, so the DePaul Symptom Questionnaire, they're using it in a way that I don't think it was intended to ever be used. They're using it sometimes, they're pulling it into research and they're using it sort of pre and post intervention. And what ends up happening when you use it that way is that you don't understand what you're seeing. And so I could be having a great week and you could do the DSQ. I mean, I'm having a great week. And so you're going to capture that I'm having I'm using quotes here, a great week. But then I could be having a terrible week and you could track that and it suddenly looks like the intervention's not working. And so I've seen researchers who are trying to do a better job, kind of pull the DSQ in, but use it in that way. And they don't hear me when I say that that's not what the DSQ is meant for. Do you have any feedback on that or any kind of suggestions?
Yeah, I mean, I have a garage at my house and I have tools in the garage, and invariably, I can't find a tool, so I'm using a different tool for the job, and it's not optimal. So I'm using a screwdriver as a chisel or something like that, and because it's just what's at hand. And I really think that, when the pandemic started, there was this huge burst of energy to understand this new disease and the chronic sequelae of this new disease. And I think people entering this research space really looked around and said, well, what do we have to work with? And we were very fortunate that the DSQ PEM was published in 2019. I mean, the timing could not have been better for us because before that, we really didn't have a quick screening tool to at least get post-exertional malaise into the conversation. But as I pointed out in my talk back in ancient history, back in ancient times in this session, it's got a purpose, and that purpose is to start a conversation. It's not to finish it, and that purpose is not necessarily to get at the disability associated with the magnitude of suffering associated with post-exertional malaise. And so we have other tools for that that we're continuing to get to know. So I would agree with you, Sabrina, that one of the things that we can do that's out of the box, would not cost any additional money, is just to use the correct tool for the job.
Okay, awesome.
Can I add to that really quick?
Yeah.
I just wanted to point out Isabel's Twitter survey. So controlling for if patients are in PEM or not. Do you want to mention, I don't remember the numbers that you got, Isabel, but it was like 50% of, or even more, patients were like, I've been in a study and I've been in PEM, and the point of the study was not meant to be measuring that. And so I think that is really important. So I just had to
I don't remember
that.
the specific results, but even just asking that question can make a tremendous difference. Yeah.
So sorry, so what would the question be that people should ask?
If when they're responding, they are in PEM and maybe even, qualifying because not all levels of PEM are the same. So, and that can give a lot of insight into what that response is based on whether or not a patient is having PEM at the time of answering.
Awesome. I would also add level of severity, really important here, because mild, I hate that name. Mild is not mild ME, but I'm going to use the term because that's the term. Mild ME can look very different as you're studying a medication intervention or some other intervention. It can look very different to have success in mild versus success in severe. And you need to be considering that. You need to be designing for that. You need to be having different measurements for that. And I just don't think we're doing a great job of even having that conversation. And I don't know that we have the perfect tools yet to be able to differentiate, because I know I glide across those lines. I'm about to glide in one direction after this conference, but a medication can pop in and I'll glide a little bit closer to that mild end, and then I'll all of a sudden it'll stop working. I'll glide back over. But when you want to test me in research, you should know where I'm at. And then you should know where I'm at the end of the intervention. So I think we do need to be having that conversation and doing some tracking with that. And Jacqueline, I was going to come to you too. I know you probably want to add on to what we were just talking about, but also the cognitive testing, right, is such a missed opportunity that we are just not doing a good enough job with. And so can you discuss a little bit about, and we've talked about this at our CDE group, and I did ask Vicki if I was allowed to mention that we're on a CDE group, but we've mentioned a little bit in the Common Data Elements group that we need to find tools that are more accessible. And so don't have a huge cost point, but we also need to start planning and investing in how we can do better. So do you want to kind of break that down a little bit, maybe, for those listening?
Yeah, I was going to agree with what everyone has said. And I also think it's not just about having the right tools, but having the right design. And that's actually something that we're not covering in our CDE group, right? Like, we're talking about the tools very much in depth, and talking about measurement and the importance of measurement, and that it's valid, sensitive, specific, reliable, etc. But the ways in which people design randomized controlled trials and the ways that they design longitudinal observational cohorts really has tremendous implications for how the data is viewed, even if you're using all the right tools. And to speak to the cognitive side of that, one of the biggest sort of pitfalls I see in randomized clinical trials for cognitive dysfunction is that, and regardless of the population, is that people they typically have so patient-reported outcomes as the primary outcome, and they include only those measures as an inclusion criteria. So in other words, patients just have to report that they have, brain fog or cognitive dysfunction in order to be included in the study, and that's what they look at post-intervention. And while I understand that a lot of people feel that that is more inclusive, I think it does patients a disservice. And the reason is because if you have the right tools, you can identify cognitive dysfunction where it exists, and you don't water down the treatment effect of the intervention by intervening on something that is already not impaired. And so what I mean by that is, an example is like for Recover Neuro. I'm one of the PIs for Recover Neuro, and the NIH was pretty strict about, making it more inclusive. And so our primary outcome measure was a self-reported measure of cognitive dysfunction. The issue that happened is that, people were reporting cognitive dysfunction. The measures at baseline were not the optimal measures to detect cognitive impairment at baseline. And so that means they were not going to be the optimal measures to detect the cognitive impairment outcomes. And so what we saw is that none of the treatments in our 5-arm multi-site trial were effective because patients were not impaired on those measures at baseline. And so that is a huge, huge, huge problem and a waste of federal dollars, frankly, when you're not able to do that. And so I think, yes, the tools are incredibly important, having the right tools, but also having the right design, right? So in other words, as an inclusion criteria, it has to be that you're capturing that impairment at baseline in order to then test whether there has been a change. And it's the same for, if you take like adherence research, right? If you want to, look at a behavioral intervention to improve, adherence to medical appointments, you're not going to recruit from medical offices, because then you're by definition capturing patients who are adherent, right, to medical appointments. And so you're not going to see a change in that regardless of the intervention, because what's the point of changing adherence from 60% to 80%? There's no clinical benefit there, right? And it's the same thing with cognition. It's the same thing with a lot of, with fatigue, with a lot of the things that we're measuring. And so having the right tools is important, but having the right design is important too. And to speak to the CDEs that we're doing, I think having a common language between researchers is hugely important. And so using the same measures, right, that we know are sensitive in the population, that we know have been tested over and over again, are really important. And that's part of what we are doing. We're choosing, measures that have been shown to capture impairment in ME specifically, and recommending that researchers use it, but they need to do so with the right design, which I think is what we're hopefully going to eventually talk about.
Yeah, and I think the one of the frustrations for patients always when they're going to a research study is that we can't get to the research study unless we rest really hard. But then we rest really hard, and guess what? We pass with flying colors. We look like our cognition is the best it's ever been. And it's because we're all jammed up on adrenaline and we're rested and we're probably caffeinated and we're probably all, electrolytes are up, like we're that's not our everyday. That doesn't show you, like you were talking about in your presentation, that's not the everyday perspective of what it looks like. So I think when we're designing research, we have to find ways to be able to reach people in their everyday at home as best we can. And I think the cognitive testing, one of the things I've learned is that some of the tools that are trying to do that are not doing a great job. So can you talk a little bit about, not to knock any of the apps, because I know there's people here using apps and I know why we're using apps. We're trying to reach people in their environment and at home. But what are some of the challenges with where we're at currently in that what are we missing?
Yeah, I mean, I would love to, I think I got this question actually after my talk, but people were asking about the most common or the best remote measures to use for trials, right? And yes, like we want that very much, but like I'm so hesitant to say, to recommend any of these, because they're just not, they don't do a great job at detecting impairment. They're typically not sensitive enough, and there's a lot of sort of error that can occur when a clinician is not present. And so you risk misclassifying somebody either as too impaired or not impaired enough. And that's a big problem, I think, with the remote testing. The other part, I think, of what you're saying is we have so you were mentioning, Sabrina, sorry, it's been a long day for me as well. You were talking about the first part. I'm trying to remember the first part because I just lost my train of thought.
Oh, I hope you don't need me to remember anything because I've forgotten everything I've said.
Oh my gosh, I just forgot what I was about to say.
I can come back. I can come back to you. It'll come back.
Yeah, I think the point of what we're saying though is just that our research really is not there in terms of having these sort of remote measures or the measures that maybe are considered, valid for certain populations, but not for others. And the ideal, of course, is that at some point we develop those, but I think really the call is for researchers to just be mindful of the fact that they will miss homebound patients if they restrict inclusion in studies to people coming in person. Ideally, we would have researchers or coordinators go to patients' homes whenever feasible and do testing in that way. And sort of the middle point of that is like remote testing through Zoom, where like someone can be present, but then you do miss certain domains that can't be tested virtually, right? And so it's sort of a big trade-off that we talk about a lot. And unfortunately, like when I tell colleagues, in the medical field that we have paper and pencil tests, and they're like, wait, you you print out paper and you use a number 2 pencil. I don't understand that. Like, why don't you have it automated, and why is it not on an iPad or using AI or whatever? And it's just not where our field is. It requires a tremendous amount of validation. It requires a tremendous amount of psychometric study to develop these tools and develop the appropriate normative data for it. And so, yeah, our field is just behind in that way. And it's hard to imagine it getting better. The other point actually that I wanted to make is just that insurance also won't cover a lot of these remote measures. And so clinically, it's very hard, I think, for people who need to see a neuropsychologist clinically and have insurance. Insurance won't cover a lot of telehealth visits a lot of the time. And so that's a big barrier as well.
Well, thank you for that. I think that's one of those
I
areas.
hope I got it.
Yeah, no,
what I was
that's
trying to
great. I
say.
think that one of the conversations we were saying earlier, there's some things that we're going to have to like get in similar rooms and have more conversation about. And I think our CDE group has started that conversation, but there's so much more that needs to be done. I think what we're discovering is the tools don't always exist, or the methods, so there's so much more work to do. So that's an area I think of exploration for ME research that we need to like invest in and spend some time.
And I just remembered the other point that we were going to talk about, which is that a lot of studies have used measures that are actually really great. So an example is like something called a digit span, which is a test of basic attention and working memory. And so that's a really great test. It's part of a very well-established battery of tests, and we use it for nearly every population. The problem with that test is that it's looking at basic attention, and in conditions like ME, that's not usually captured because the deficit is not in basic attention, it's in sustained attention over time. And so in a study, they'll say like, oh, we need to include a measure of attention. Here is the digit span, right? And then they'll say, well, people with ME are not impaired in attention. But so it just speaks to the importance of using the correct tools and getting at the correct subdomains, which I alluded to a little bit in my talk. I remembered. I came back to it.
Good job. Okay, well, that's great. Michael, I want to come to you and just ask you, because I know you've been in this space you've been in this space a long time. You've been in the space pre the pandemic. And so you've seen a lot of transition, a lot of ideas probably come and go. One of the areas I think patients are still really keen to see more research is on the brain. I think we're really excited about the idea that somebody can visualize and explain some of what we're going through and what they can see, so that I think it's because it holds so much credibility and validity that in many ways, because we're struggling to be seen as valid by clinicians, it gives us this tool where we can be like, look. And so I guess, from your perspective, given that you have so much experience, what are you seeing that's exciting? What are you hopeful about? What do you think we could be doing better at in terms of moving towards doing more research in that area and better research in that area?
Yeah, I mean, I guess one problem is that I surely can't do home visits with our, $2 million dual PET MR scanner. It's just a hard problem, right? So it naturally selects for certain participants. Our participants are really generous in coming in, but they must come in for our stuff. So what we try to do is to get measures from our study participants that can come in, and we also do at-home phlebotomy, where, at least at this point, just in long COVID, but we are working on ME, where we have a little, funding from a donor, so they say what they want the money to go to, but we have set up an infrastructure to collect blood from at-home participants. So we can do the same, the neutrophils that I showed in the talk. We do that from at-home participants and in-person scan participants, so that hopefully we can weigh in a little bit on what might be happening at home if we have a good correlation. So, I mean, I agree that there's a certain credibility that happens with neuroimaging. I don't necessarily think that's great, because sometimes I feel like we're studying smoke and there may be fire. And I think a lot of people are studying smoke. It's not necessarily the case that we're studying fire. But there is a credibility that it's just true. Like, people like it, doctors like it, media likes it. That's part of the reason that I do it. I do think the stuff that we do is valuable and important, but I think sometimes it's outweighed. If I could, I would be doing, you know, like gut biopsies on people and things like that to get into tissues and try to figure out what's going on and that sort of thing. But yeah, I mean, I think there's good research, and from my perspective, what we try to do is to combine, because a huge combined methodology is a huge problem in this field and others, is that everybody has their own sort of siloed expertise and they don't get other samples. So what we try to do is to get multiple measurements from people before they get in the scanner. And then we will ship out samples of blood, saliva, et cetera, to collaborators to try and weigh in on stuff that I can't do in my lab. And we even get, let's say, for example, in small fiber neuropathy patients, we get skin punch biopsies from patients who have also undergone neuroimaging. So we start to connect modalities. You know, I think a lot of people that gave talks made the same point over and over again, which is that there's a lot of different things that can kind of go wrong. There isn't some phenotype that's just everybody. And I think that collecting across modalities is something that can be helpful in all labs, but particularly in neuroimaging labs, where at some point it's not all that helpful when someone says, you know, I have a lot of trouble concentrating and I can't focus and I get worn out. And I go, oh yes, my $1 million machine agrees. You know, okay, that's cool. I guess there's a validation and we can get at mechanism, but I think combining modalities can be really helpful.
I think you've always believed patients. That's why for you, it's probably not something that you think you need to do. But for us, we're up against so many people who aren't like you that when you say something, I run to my doctor with it. I literally print it off.
That is literally
Look.
part of the reason that I tried to penetrate Harvard Medical School and get into these places. You know, I have a very sick friend that I grew up with that I just think about all the time. I talk to her all the time. And I just thought that it shouldn't be this way, but it is, that I will just have more credibility if I can penetrate some of these institutions and use fancy technologies and that sort of thing. It's just the truth.
Well, thank you so much for everything you've done for us. You've been here since long before there was money to do half the stuff that people are currently doing now, you know, so fortunately. So we appreciate everybody who's here before and who's here currently and has come in due to the pandemic. However you got here, we're happy you're here. Dr. Moreau, you have sent your team into homes to try and engage the more severe population, which is a big priority for our community. We've always said we want to have more severe patients in because we think one of the problems in our research is that when you can only see the milder version of the patient, you're getting sort of one perspective on what the illness looks like. And that isn't always indicative of sort of across the board or what it's going to look like to treat that or what it's going to look when you're providing supports to that. And so we have for a long time been asking for researchers to think about how they can do more things with more severe end patients. And you are doing it. So
exactly.
any great lessons you've learned? And I mean, do you feel like it's given you a stronger signal to study some of the things that you're looking at?
Yes, two reasons. So first of all, the home-based protocol is over a week. So there are 3 visits of the clinical research nurse. And we have the first 2 days to establish the baseline value. We are recording our deep phenotyping protocol is quite, I would say, exceptional. Okay, it's a bit costly. Okay, but thanks to OMF and private donors, we can afford that for the ME patient. But we ask them to wear the exoskin connected vest, so at least 20 hours over 24. Trust me, they are wearing in average 23 point something, so the something is maybe the shower or something like that. We ask them to have to wear the vest for the whole night, okay, to record sleep disturbances, because there is an algorithm special developed by Kari Technology for that, their vests. But we have heart rate, respiration, breathing rate variation. So when they are tachycardic at home, I know it. We saw exceptional things. They are not tachycardic because they are going to visit the doctor or in the waiting room. They are tachycardic at home in their own environment, which is a game changer. We're collecting saliva, first morning urine every day. So we know how on the metabolic point of view, we know how exactly the PEM is developing after we use a cuff to induce PEM. I know a lot of people are quite suspicious about whether the cough is enough. First of all, this is a cohort of very sick patient.
Are we suspicious or are we just still learning?
Challenging. Okay, let's say
Challenging.
the
All right, I'll
wording
use that.
challenging, which is okay, But let's say that we are doing all the omics possible for that, which allowed us to stratify patient. For instance, we have up to over 40% of patients coming to us with a medical diagnosis of ME, which have some symptom overlap with ME, but when we test them with our home-based tests, they are FM, pure FM, okay? And we have other markers that are purely associated with FM confirming that. So we are this unique capacity to further screen the patient and differentiate them, not only to assess severe one, but also to look at them with different lens. And when you do your omic, it's easy. I have over 600 patients in my biobank and data set. But when we start, even with the first, let's say 200s, we were enough power to see things that nobody else have seen because we have this more homogeneous subgrouping of patients. I remember my very first talk in Stanford several years ago, that the question of the day was, are you a lumper or a splitter? And I was tagged splitter, and I remain splitter, okay? But I don't think it's a bad thing to be a splitter because this is where I introduce precision medicine. I'm not splitting to avoid some patient. I want to treat them with the best efficient drugs or the way that we can safely address their needs. And nobody will know all the stone in my garden are turned, and I turn them many times a day. So that's why we are collecting. We are measuring brain oximetry change before, during, and 90 minutes of the stress test and after. Nobody is doing that. So we are using portable machines that are not that expensive. Of course, I would like to have access to an MRI or other CT scan to further complement that, but that will be impossible with this cohort of patients. And we are collecting that. So for instance, for Dr. Baker, we are using brain check tests at baseline immediately after the stress test and 5 days later. You will see soon our first paper. Agree or not, we have sensitivity and we're seeing things, okay? That we are now matching with circulating factor that are connected to cognition, which make a lot of sense to me. So I'm the first to challenging our own findings. Okay, I'm not saying that everything we are touching will transform in gold, which is not true. And also to piggyback on what Michael and Todd said before, I'm just a simple tool man addressing complex disease. I'm not a proteomic guy. I'm not a metabolomic guy. I'm not a transcriptomic, okay? But in my toolbox, I have all those omics, and I don't care which one will give me the answer to improve the life of people living with ME. So I don't have this curse of the expert, you know. I heard so many colleagues, and I repeat that even at the NIH, I don't care. I don't think that a proteomic guy will fix the whole issue with proteomics. Same with the metabolic. I have dear friends that are top expert with each of these domains, okay? I need them. But at the end of the day, I'm just a tool man, and I like to use all the tools available, even those that I'm discovering or I'm improving a bit. But I'm a simply tool man.
Well, I'm going to put that on your bio from now on. I'm going to put Challenger under mine, and I'm going to put Toolman under yours.
Yeah.
Alain and I have rich conversations about these kinds of topics, and he's very good to have those rich conversations. And he loves having them, despite what he may be implying at the moment. One of the things Alain brings up that's really important is provocation methods. So that's an area that we know we need to do more research in as well, as provocation methods means how do we provoke PEM? And are there better ways to provoke PEM that we could test against existing technology like the CPET? And if we in fact find that something does the same job as the CPET, then we could let the CPET go and we could move towards that tool. And I know we heard earlier that, you know, some people are letting the CPET go already, which, you know, everybody has to figure out kind of where they're at in terms of what they're using, and that's on them. But for me, one of the things that has always concerned me is that we haven't had enough conversation about what those tools could be. And, you know, when I challenged Dr. Moreau about the provocation method, I have a similar tool at home that doesn't cause PEM. So I've just, you know, that's the batting around of like patients talking to researchers and clinicians, is like sometimes you can kind of strengthen an idea or a thought. But if that cuff in fact is measured against CPET and was to be found to be the same, then that would be a much less invasive way of provoking PEM. And I'd really love to see, and Jacqueline and I have talked about this, you know, a cognitive test that could provoke PEM. I mean, I can't read a few pages of a book without putting myself into PEM. So we know that we can create that same thing. How? I don't know. I'm not a brain expert. That's what we have all these brilliant people here for. But I know that books crash me and I can't read more than a few pages. So that tells me there must be something we could do cognitively that would become a test that could be less invasive than me having to get up, leave my home, and go and do something really tough like a 2-day test. So for me, that's just another area. We're not going to solve it today, but it's another area to put on the radar of like, we need better provocation methods, and we need to get in a room and start talking about what we have, how to test what we have, how to think about new things we want. It's going to take years, but that's okay because we have years and we have to have those tough conversations. I think this idea that ME is going to be fixed tomorrow, we need to like, I've heard so many people say, well, there's no point in figuring that out because, you know, we're so close to this. Like, I've been in this for 10 years now. We have time to go and create some of these tools. And by God, if somebody discovers and cures it all tomorrow, we'll drop everything we were doing and it'll all be fine. But if not, we'll have continued to move in the right direction with some of these things. So we've mentioned a couple of areas: PEM, provocation tools, there's cognitive testing. There's definitely more work to do in some of those. We've talked about patient engagement and accessibility in research and how important it is. We've talked about educating clinicians and how important that is. And ICAN-CME is trying to do as many of those things as we can. We always need new partners. We always need more people getting involved. So by all means, if anything you've heard in the last 3 days has piqued your interest and you want to partner, give us 2 weeks because we need to rest. But then by all means, reach out, because there are opportunities at ICAN-CME always to partner and to collaborate. And with that, I just want to ask if anybody has any last thoughts as we kind of go around, and then we'll do a quick wrap up of the conference. But are there any final thoughts? Anything we didn't talk about today that you feel is really sort of pressing? Anything you want to clarify? I have just one thing. The framing, I know, Alain's laughing. Just the framing of what you guys think about it. Just the framing of how we think about each infection that brings people into the ME world. I think we have to be really cautious about how we're framing the language around that, because we have all of these people in our community who've come to us sometimes through unknown infections and sometimes through infections that we knew. And they've had different experiences sometimes. If they came in through COVID, it was a very different experience than maybe if we came in through EBV or something else. Everybody matters. Everybody's important. Everybody deserves research and care. Not everybody's having the same experience, unfortunately, but I do think that when we frame sort of one set of patients versus another set of patients, my ask to you, and I hear this from patients all the time, we don't want to be divided. If there are people who want to divide themselves, that's okay. I respect their autonomy to go and feel how they want to feel. But generally speaking, what I would love is if we could talk about the differences that we see in a way that feels just a little bit more respectful of the fact that everybody is really important and matters. And I say that only because sometimes I'll hear people say, well, this is different, so therefore more important, and I don't even know sometimes if they're saying it or if it's just implied, but I know that I sit there sometimes and I feel like, like, as my version of this not important enough? And then I talk to patients and they've all had the same reaction. And I think it's so easy to just frame the conversation differently. Every infection matters, and every infection is going to have a unique signature, right? That's the way I say it to people. There's going to be unique things that are going to come with every infection. I don't know what they are. I'm not smart enough, but I know that there's really smart people working on that. But whatever they're going to find is going to be really useful to that group that came in through that infection and knew that they came in through that infection. But there's a whole group of people who are never going to know the name of the infection because we almost do no testing. Doctors hardly want to see us. They tell us to stay home and have some chicken soup and hope to God it goes away. And we start catching infections from the time we're in the womb. So like when people say to me, no, an infection didn't cause it, and I'm always like, well, how do you know? We've been having infections our whole lives that we just thought were harmless, and maybe we were wrong. Like, so I guess it's just a plea to just, when we talk about these illnesses and we're subdividing them in the research, to just be mindful that we're real people getting subdivided into categories, and we all want to matter, and we all want to be fixed and cured and get treatments, and we all want the other groups too. Like, I'm not begrudging anybody their moment. I want COVID to be solved and figured out. I think it's been given a lot of money and a lot of support, and if we figure that out, we probably figure some of the other stuff out. But just don't forget us too. That's all. And that's my final word. And now I come back to you guys. Do you guys have any final words? How could you follow that, really, though? Would anybody like to share anything as we leave this wonderful time together? Oh, nothing. Okay. All right. Well, we can social media it later if anything pops into your mind. Alain, did you want to say a few final words of thanks to everybody? And just in terms of, we're ending the conference now, and this is our last few minutes together, and I know you'd like to say a few words.
Yes, thank you very much, Sabrina, because as I said initially 2 days ago, you're already the heart and soul of this network. Okay, we work very hard. We have a long journey so far, okay, starting in 2018, and we work and we develop some kind of consensus. And there's a big difference between iCanCME 1.0 version versus the 2.0 version, where the 2.0 version, we really create this vision that patients are partners, patients are leaders, in fact, co-lead the network from A to Z. And eventually, in a few months, I will step down as per my role of director, okay? But we will agree on the way that we will proceed for that. And it's not because I'm tired and I will do something else, trust me, okay? But is it my own view as research leaders that we need to renew leadership in network. We need to renew leadership in different organizations. I do believe in that very much. It's also my duty to make sure that whoever will step in, and I have no doubt about that person, how can I will help these persons to progress, to participate also in the writing of iCanCME 2.0, as I did before in another research network on oral health. And to make sure that it's not because I will let someone else to drive. Okay, I will remain a very active participant, but I think it's naturally to do that. So thank you, Sabrina, for your contribution. In fact, outstanding contribution as always. And you will see in the very next seconds in your email, check your email. Okay, there is a small token of our appreciation, okay, from on behalf of the member of iCanCME Research Network to acknowledge your outstanding contribution in the organization and everything you have done so far for this outstanding conference. Okay, thank you. Also, there will be a recognition package for all the organizing committee members, okay, from iCanCME, okay. So you will be in touch next week. Check your email. It's not a check, but it's something equivalent, okay. So we are taking care of you because, again, and that's been mentioned today, it's something very important that we recognize all the energy, time, and not only effort, but expertise, because each of you have unique expertise that you contribute to this network, okay? And this network is interacting with other network. Today we have Renegade Research, and it's interesting because I have a new collaboration with Isabel as a patient partner on a new project that we submit to Solvimi. Finger crossed. We'll see what happens. Okay, but that's the way the things are evolving. So again, my heartfelt thanks to all of you. Sabrina, please rest, enjoy the rest of tomorrow and after tomorrow. Okay, you deserve it, but also my most sincere thanks to everyone. Okay, you did a great job. I like very much Carrie, and I think Air Canada should hire you to do some bilingual message in the airplane and flights. You do a great job. So thank you all, and thank you for our technical support also, who also play a very different role. I've been in this type of webinar in many times through different organization. I'm quite biased here because this is from I Can't See Me, but I saw the high quality of the question from the audience. I hope you appreciate the sharp question. I was very busy to answer myself and very sharp questions. So stay tuned for maybe a next conference, not next week for sure. So thank you all, and thank you for all our speakers. Okay, you did a great contribution to the success of this conference. Thank you all.
Yeah, and thank you to the planning committee for being in the background and just making all the little things that were supposed to happen, because there's about 8 million things happening in the back end of this. I just get to be the face, but there's lots happening in the back end, and our team has been really amazing. And it's taken a year and a half to get here, so an acknowledgement of all the people who've come along the way. And especially in the last few weeks of panic, a lot of people jumped in and helped us with technology fixes and things that we really appreciate. So thank you to the speakers, thank you to the attendees, and thank you especially to the planning team. And you're going to hear from us because we're going to do more of this stuff. See you soon. And fill out the survey. Bye.